So, dear colleagues, ladies and gentlemen, welcome to this workshop. I'm honored to welcome so many industry experts and colleagues from academia and regulatory agencies and other organizations to the EMA for this afternoon. I think this is particularly important for us who have been involved since many years in setting up the Cancer Medicines Forum together with the RTC. We've always said at some point we want to reach out to our stakeholders, and this is a first step in this direction. Now, why it is so important is because there is such unprecedented development in clinical oncology, but at the same time, the speed by which drugs are brought to the market to address high unmet needs also leaves a lot of knowledge uncertainty in some areas, and that's why treatment optimization research is there to answer some of these questions. Today, I want to focus on perhaps the most challenging type of research, which is the clinical trials. Often these are not only very challenging for scientific reasons, the research questions are complex, but also for practical reasons. These are trials that are difficult to conduct. Now, I'm not claiming that we will be able to find quick solutions to complex issues. What I would like to get from today is an understanding of everybody's will and commitment to address these problems, not just through discussion, but through a collective effort. And I think with our colleagues from the Cancer Medicines Forum, we have a place now where we can bring together so many stakeholders, and we want to really explore whether we can extend this collaboration to industry stakeholders. So while we won't have a lot of time to address all of the issues, we've designed a program which addresses at least some example of key issues that we could be working on. And they have been designed mainly for the speakers to tell us their insight about some of the problems, not so much for discussion. We will leave the discussion towards the end of the meeting. Now, I will be quite rigorous with the timing because we have a lot of speakers, a lot of very important presentations, and we want to give justice to all of them. So while I'm not going to be interrupting the speakers, at least not in the room, I will signal you when there are a few minutes left, and please, there is a need to conclude. At the end of the day, when we have our panel, I want to leave this meeting with a clear understanding of whether there is a common ambition to work together and facilitate optimization trials, and what you think, you as industry or whichever hat you're wearing at this meeting, what you think EMA can do to help in this. So to kick off the discussion, I'm pleased to introduce our first speakers, Michael Zajac and Domnita Ileana Burcoveanu. Michael occupies a particular place in the list of speakers because he has helped us design the program and has been an observer in the Cancer Medicines Forum for the industry. So without further ado, the floor is yours.
I hope, yeah, it works. So, thank you. I'm Domnita Ileana Burcoveanu, and I'm a medical oncologist by training and joined pharma 5 years ago. I'm currently medical affairs with Bayer. Next slide, please. So the years that we have spent in drug development have taught us that registration trials are not necessarily reflected in the clinical practice, because the patients are not the same, the clinical settings are not the same. So we came to the conclusion that after the math, after approval, often we will need the clinical practice experience from the doctors, patient view, payers view, and also the health system perspective in order to optimize the sequencing, the duration, and sometimes even the dose. And this can be and should be done by pragmatic, adaptive, living trials where we can really assess the real-world risk-benefit. And in case that dose modification is needed, it should be clear who, why, more individualization of the dose than really just altogether modification. Because while we need to be sure that the patients are safe, we also need to make sure that the efficacy is maintained. Like Churchill was saying, live today and you'll be able to fight tomorrow. Michael?
Then I give a short account of the first 3 years of CMF, and I think the next 3 slides have a lot of detail, so they're in the pre-reads, so please take them as a pre-read. But the first slide sort of is forming. I think we formed in 2022, and I think the prime goal is and was optimization questions in the post-licensing session. I think throughout the discussions, we also come, of course, that the same methodology and the cooperation could also apply, of course, in the pre-licensing setting. But our focus truly is the post-licensing setting. And I guess after a year of forming, we went sort of into a more storming period, and it doesn't storm. The slides don't go. Now it goes. Okay, excellent. So we started to consolidate. We had a lot of external advice. We had a lot of external presentation from academics, from peers, from regulatory colleagues, to really look at what is the expectation, what's the terminology around the trials. Dominika mentioned pragmatic trials, adaptive trials. We all use different terminology. And I think we sort of settled on where we want to go. And that culminated in the first workshop we had. That was in April 2024 in this room here, where we really confirmed our 2 goals, which is the identify opportunities for optimization. And that's where Francesco, I think, pointed to, is that's what we really wanted to end up with today as well. But also start looking to facilitate the delivery of these kind of projects. So with that, for me, I think it is a great experience to observe the Cancer Medicine Forum. And when I do that, I often think of another Churchill quote, which is he made on the eve of the Battle of El Alamein. He said, "This is not the end. It's not even the beginning of the end, but it might just be the end of the beginning," which means we are really on a good track, but we need all of you in the room really to work together. And with that, I give back to Iljana.
So what would be potential future concrete direction, actually? It would be important to normalize optimization as integrative part of compounds' life cycle to maximize efficacy, minimize toxicity, but also spare time and resources in general into making innovation available to patients. Another possible direction would be participative participating approaches in engaging as early as possible in drugs life, cross-disciplinary consultations and expertise that may improve clinical trials design and therefore their outcomes. Also, it would be interesting to explore innovating funding methods that facilitate collaboration between industry, of course, but also public agencies, academia, and the payers. And last and not least, we already mentioned that at least twice today, adaptive trials that use shared dynamic databases and platforms. And also, a wider use of real-world evidence would be important into informing optimization. Next slide, please. So, in our view, my colleagues and I, we are moving from theory to practice. So we need to actually focus on specific topics to see how we can get to pragmatic European trials that go out with the data that the payers may require swiftly. And as mentioned, as early as possible, integration of optimization into the drug development. For us as industry, it would be an opportunity to minimize the risks that is inherent to any drug development, especially to maximize the success possibility to get approval and then access to market, right? And stabilize the access and to improve patient outcome. Because at the end of every day that we are clinicians, researchers, payers, regulatories, we are all here to make sure that patients are best taken care of as possible. And with that, I pass it to Francesco, the first speaker.
Thank you very much, very clear, and thank you for being perfectly on time. So, colleagues, I would move on to our first session for today, finding the optimal dose and duration, an ongoing challenge. Our first two speakers are Chunze Lee and Leslie Carter. The floor is yours.
Yeah, thank you, Francesca and the organizing committee for the kind invitation. It is our pleasure to be here. So first, I just want to highlight Pavan from AstraZeneca and Leslie from AbbVie. We are working together for this deck. So the presentation of my title is a challenge and opportunity for the oncology dose optimization, the industry perspective. Next slide, please. So here is the quick outline of my presentation. So first, I would like to give you a brief introduction on oncology dose optimization, why it matters now, and then highlight some of the key challenges and opportunity with a case examples. Lastly is a summary. Next slide, please. So the concept of oncology dose finding has evolved with the new therapies. As you can show this slide on the left, for the traditional chemotherapies, the paradigm was simple. The efficacy and the safety are highly correlated. So we escalated the dose to find the maximum tolerated dose, which is MTD, which ultimately driven by the DLT. And this MTD are usually selected as a recommended phase 2 dose. So can you have click twice? However, these MTD paradigms are often not applicable for the modern target and immunotherapies. And with this newer agent, you can see the dose response curve, the therapeutic window are changing. The higher dose does not necessarily result into the greater anti-tumor effect. The optimal biological dose, the OBD, can become more relevant. So as you can see, as the flatter dose response curve for the target therapy and the cancer immunotherapy show, the DLT alone is no longer sufficient to guide our dose decisions. We often need to select RP2D dose that are not MTD and require us to consider both safety and efficacy signals early in the development. Next slide. So did lead to a new framework for those findings. It's no longer a simple linear decisions. It's a multiple dimensional optimization problems. We have to balance a wide variety of the factors, not just acute safety and efficacy, but also PK/PD, long-term tolerability, and the patient report outcome, etc. And those decisions now is a complex benefit risk profile assessment across multiple dose levels. Next slide, please. So the new framework presents a significant challenge. So first, our first-in-human studies has multiple interconnected objectives. So we try to finding the RP2D dose. Besides that, we also try to find MTD, understanding the patient population, and finding demonstrate POM, POA, etc., all at once. So we ask a lot from our first-in-human studies. Second, we also face significant ethnic constraints. It is not ethical to expose a large number of patients to a poorly tolerated or suboptimal dose for this cancer patient. And the biological and the patient heterogeneity also make establishing a clear, robust dose exposure response relationship very challenging in early clinical trials. Furthermore, our early clinical studies have shorter observation windows, which limit our understanding for the long-term tolerabilities and also even long-term efficacy benefit. And finally, I also want to highlight we often lack reliable and clinical relevant biomarker for efficacy, and additionally, our early efficacy endpoints does not always reflect the long-term benefits like survivals. So as a result, the dose finding and dose decision can be very complex and context-driven, and the dose decision often need to trade off and balance multiple factors. So as a result, the dose decision based on benefit risk profiles can be very subjective. Next slide, please. Despite of these challenges, there's also a lot of opportunities for us to improve oncology dose findings. So here I want to briefly highlight the 4 key areas. So the first one is a tailored dose optimization. So we are moving away from one-size-fits-all approach and tailor our dose finding strategies by molecular class and mode of actions. Second is innovative study design, using model assist algorithms and adaptive study design to further enhance robust dose findings. And third one is modeling and simulation by integrating the totality evidence to guide those decisions. And lastly, not the least, is the collaboration. So I would like to highlight the enhanced collaboration and communication across industry, health authorities, HTA, and the payers, and even academic is quite crucial to be successful to further improve those optimizations. So due to the limited time, I will briefly touch upon the first 3 topics with the illustration examples. Next slide, please. The first opportunity is to tailor our dose optimization approach. We have identified 4 distinct molecular classes based on their expected single agent activity and the mode of actions. Class 1 is a small molecule and antibody drug conjugate. Class 2 is a large molecule antagonist like pembrolizumab. And the class 3 is cancer immunotherapy agonists like T-cell engagers. And the class 4 is the most challenging one, is a drug with limited or no single agent activities. So next slide, please. So while a very generic clinical development plan apply across classes, each class also require unique consideration to optimize those optimizations. For example, for class 1, the small molecule and the ADC, we know the preclinical animal models are often more predictive, and we can effectively leverage this information to guide those finding strategies. On the other hand, for class 2 molecule, the large molecule antagonist, we can potentially leverage target engagement data to support those selections. So it may not make sense to continue dose escalate if you have achieved full target engagement at a set of actions. And for the class 3, the CIT agonist, for example, T-cell engager, we may need to consider optimize both step-up dosing and the target dose. And then lastly, for the class 4, where there's no single agent activity, the strategy is to move as quickly as possible into combination studies to demonstrate the proof of activities. So next slide, please. The second opportunity in a novel study design. We are moving away from simple rule-based design like 3+3 to more sophisticated to model-assisted algorithms such as Bayesian, Effitox, and DROD, allow us to incorporate both toxicity and efficacy data together to make a real-time dose decisions. And this enable more robust and efficient dose finding and help us identify accepted dose range based on totality evidence rather than DLTs. Additionally, I think there's many publications to show that even for the dose finding, we need to identify clear dose decision rules before the study start, such as decision tree, utility analysis, by incorporating both safety and efficacy of merit algorithms. Next slide, please. The third opportunity I would like to highlight is the modeling and the simulation tools. And these slides just show a wide range of tools are available to us, ranging from POP-PK, PBPK modeling to QSP and explorer response analysis. And these models are essential for integrate all the preclinical and clinical data together to enable robust real-time dose decisions. Next slide, please. So now I just want to use polatuzumab vedotin as a case example to illustrate how we apply these principles. So again, here the graph on the we are showing here is the explorer response analysis for polatuzumab vedotin treatment up to progression for both efficacy, the objective response rate, and the safety. In this case, is a peripheral neuropathy. As you can see here, we see a positive ER relationship for both safety and efficacy. Another click, maybe couple. Thank you. But however, when we cap the treatment duration up to 8 cycles, you can see the ER relationship for efficacy remain the same, while the ER safety, the trend is a little bit flatter, indicating when we cap the treatment duration, we may potentially risk mitigate safety while maintain the efficacy. And additionally, time to event model showing here confirm that 1.8 mg per kg capping up to 8 cycles will strike the right balance between efficacy and safety. Another bill. And this dose regimen ultimately leading to a successful phase 2 studies, which demonstrate strong efficacy in relapsed refractory DLBCL, and ultimately leading to the global approval for these molecules. Next slide. So here I would like to highlight dose oncology dose optimization is a continuous journey. It does not end as a phase 3. Even after the approval, we often continue to explore alternative dosing and schedule to enhance the patient convenience and further improve therapeutic windows. As this graph shows, those findings is a continuous learning and confirming process. As we gather more data from preclinical all the way to phase 3, even phase 4, our confidence in the dose selection is increased and uncertainties are gradually decreased. Next slide. So in summary, dose optimization strategy should be seamlessly integrated to the entirety, the end-to-end clinical development plan across life cycles. So today I highlight some of the tailored dosing strategy based on molecular class, leveraging novel study designs and the modeling and simulation to integrate totality of the data to support those decisions. And also it's important to note from industry perspective, we strongly prefer to optimize the dose before starting the phase 3 studies. As you can imagine, other speakers also mentioned, changing a dose post-approval will bring tremendous commercial, regulatory, and logistic complications. Looking to the future, we also see great potential in the novel biomarkers like ctDNA, the use of real-world evidence, AI machine learning tools to further optimize study design and perhaps even enabling personalized dosing. Next slide, please. So finally, I just want to acknowledge my colleagues at the Roche Oncology Dose Optimization working groups who contribute to these works, and the Polivi project teams, and also Genentech Clean Farm. So with that, I would like to thank you for your attention and more. Yeah.
Thank you very much,
Yeah.
Chunze. And in the preparation of this discussion, I just need to note that Leslie Carter and Pavan Vajja also contributed. Pavan, did you want to add something? No.
And we can discuss in the panel discussion.
Okay, perfect. Thank you. So the next speaker is Olli Tennunen. Should dose finding be concluded pre or post-approval?
Okay, thank you very much, and thanks for the invitation. A regulator's view on the same topic, whether dose should be and what should be done before authorization and what should be done afterwards. These are my disclosures, so I'm mainly speaking here as an individual rather than from the official point of view of the agency. So I also started my career by teaching pharmacology to the medical students, and as mentioned by the previous speaker, this was how drug development in oncology was done 20 or 30 years ago. We all know the story of identifying the MTD and recommended phase 2 dose, and then continuing to the phase 3 and then finding the optimal dose in real life, and how dose escalation with chemotherapy was done, and the therapeutic index defined. This was the era of chemotherapy, but to what extent is it applicable today is a different question. Just to say that maybe it wasn't easy even at that time. And the issue itself is not really new, is it? Etoposide was synthesized in the 60s, and the chemotherapy handbook of today says that you can use it in a dose range of 40 to 200 milligrams per square meter, days 1 to 3 or 4 or 5. And oxaliplatin, which was authorized 30 years ago, whether we should use it at the 85 milligram dose every 2 weeks or 130 every 3 weeks, do we still know that? Should we give it for 6 months in the adjuvant colorectal setting or 3 months? We don't know that. So the issue is not really new. Or how accurate is the dose finding and administering these drugs in reality? How accurate is it at all, considering the way the body surface area was originally defined based on a couple of dead bodies in the early 19th century. But it does get more complex with the products of today, of course. When you ask this type of a question from a regulator, we always, as the first step, we of course look at the legislation. Does it give an answer to the question? And this is what the European current legislation says, that the marketing authorization is granted based on properly or sufficiently demonstrated quality, safety, and efficacy of the medicinal product. And elsewhere, it says that the marketing authorization can be shall be refused if the medicinal product is harmful, or therapeutic efficacy is lacking, or is insufficiently substantiated. But does it say something specifically about the dose? No, not really. And in a way, we of course tend to think that the BR, benefit risk, is something absolute. The benefits, if they outweigh the risks, the drug should be authorized. And here comes the gray area. The dose, on one hand, it does, in most cases, it affects the safety of the drug, it affects the efficacy of the drug, and not only these, the adherence to the drug, quality of life, and ultimately to many aspects which are also incorporated in the benefit risk assessment. The next place to find an answer for a regulatory question is, of course, the guidelines, and this is what the old ICH guidelines says about the dose. Assessment of dose response should be an integral component of drug development, meaning in this case probably the pre-authorization setting. But then comes, one of my favorite sentences in drug regulation: even well-laid plans are not invariably successful. And at the time of authorization, you might be faced with uncertainties. And even though if you have uncertainties, if you have an acceptable balance of observed undesired effects and beneficial effects, you should authorize the drug. But this may lead to a situation where further dose finding might be pursued in the post-marketing period. So the guidance doesn't specifically say that it should be done in the post-authorization setting. It says that it might be pursued. Going now into the more details, I won't go into the details of the guidance in terms of oncology. You all know these guidelines, our anti-cancer guideline in Europe, and we actually do have quite some detailed guidance in the text, considering both cytotoxic compounds and non-cytotoxic compounds. And this, I think, is quite well in line what was said by the previous speaker concerning different types of anti-cancer medicinal products and how they should be approached. And the FDA have published their new guidance, as you probably know, as well as a part of the Project Optimus they've had. And this highlights the pre-authorization part of dose optimization and the benefits that might be there if you do it in the pre-authorization setting. So, as being said, in the context of today's anti-cancer medicinal products, the MTD concept doesn't work that well, and neither does dose-limiting toxicity on a conceptual level. On the other hand, the MTD as a concept, it never reflects low-grade toxicities or the duration or no dosage modifications that need to be made in the clinical practice and so on. So in a way, the MTD as a concept, even in the context of chemotherapy, doesn't answer. It leaves multiple questions unanswered. And this, of course, would favor dose optimization studies in the pre-authorization setting, even though I think we have to all admit that as a study that a classical MTD study is kind of a clear approach to the problem with simple decision rules, and by definition, the dose optimization pre-authorization trials are multidimensional and more complex, obviously, because they try to address multiple questions: the safety, adherence, clinical benefit, and so on. Having said that, I don't think the dose optimization strategy in the pre-authorization setting, it doesn't really preclude seamless designs or adaptive designs or this type of modern approaches. In a way, there are pros and cons. You can gain more with doing a proper or a multidimensional dose finding in the pre-authorization setting, but it is more complex, of course, by definition. So the FDA, as part of their Project Optimus, they've analyzed their decisions and marketing authorization assessments, and they reported in the JCO that the 15% of their approvals had a post-marketing requirement for further dose optimization. Is this a high percentage? I mean, genuinely, the uncertainties may be there even in higher percentages than 15%, but perhaps that's not the real concern there as much as is the second highlighted point on this slide, they noted that it took 6 years in median to fulfill these post-marketing requirements in the post-authorization setting. And obviously, 6 years is a long time to use a drug with a, I wouldn't say a wrong dose, but with a suboptimal dose. So there is obviously, well, it depends on the point of view, whether this is something that would favor doing it in the pre-authorization setting, or is this something that would favor to find new ways and incentives to make it more efficient and faster in the post-authorization setting. Okay, well, we know there are obvious challenges with the post-authorization studies. We will address these in later on during the following presentations today. The recruitment issues, funding, I won't go into the details. Having said that, it's rather obvious that in the pre-authorization trials, you cannot address everything that you will ultimately need in the optimal use of an anti-cancer drug in the clinical setting. You cannot have sufficient, or let's say full data in all relevant subgroups. To have a sufficient number of older adults, whether you should use a lower dose in them, whether you should use a lower dose in frail patients. There's always a learning curve in the post-authorization setting, and something is always left in the post-authorization phase. I had a few examples. In the interest of time, again, I won't go into the details. The first one here is to highlight that sometimes, indeed, it does make a difference in the pre-authorization setting. T-DXd, a well-known ADC, with 6.4 milligrams per kg versus 5.4 milligrams per kg. Numerically, very small difference, but for the grade 3 adverse events, 58% versus 38%. So you may speculate that the clinical development of this product would have been rather different if you hadn't done the dose optimization in the pre-authorization setting. But indeed, it gets more complicated with drugs which do not have a direct dose response, at least above a certain dose range. The initial dose-finding studies of pembrolizumab, dose range of 0.05 to 10 milligrams with no dose response of above 1 milligram per kg. How do you do a dose optimization in the pre-authorization setting while obviously you have a major innovation at your hand? A very complex issue. And a third example where probably, even though if you do a post-authorization randomized dose-finding study, not everybody is happy with the regulatory conclusions of that, but I won't go into the details of this complex example either. The new legislation, we won't know how it looks like in terms of this issue. It's not yet there. You may somehow read between the lines that there is this issue has been discussed there, and there are some aspects which emphasize the role of the post-authorization commitments and studies, perhaps more than the current legislation, but we don't know yet how this will ultimately look like. So with that, to the question, I don't think there's a one-size-fits-all answer. The important thing is to identify the dose related uncertainties, identify them early in development, and understand all the clinical aspects the dose might ultimately affect: the safety of the product, adherence, clinical benefit, potentially even subsequent treatments, quality of life, overall survival. So those aspects that are included somehow in the benefit risk assessment as well. Don't forget the pharmacology. The drugs are genuinely different based on their mechanism of action. Irrespective of whether you do it in the pre-authorization or post-authorization, do it prospectively, plan it prospectively from the early on for optimization. And do it by keeping the obvious challenges that would favor doing more in the pre-authorization setting. Some things are easier to do in the pre-authorization setting than in the post-authorization setting, recognizing the challenges there. And also you can learn something from the history of oncology as well. With that, I will conclude. Thank you.
Thanks a lot. All the excellent examples and showing really the dilemma that we have here. On one side, let's say we strive to optimize dose, but for many drugs, this is an enterprise that takes many decades, if it's ever achievable. Perhaps we need to lower our expectation. Luckily, as we've seen, we've got some rational strategies on how to approach the problem. Shunze presented one approach, which is very, let's say, mature. But ultimately, like Michael and Domita said, it's a matter of making this an integral part of the development plans. Whether it's before or after will depend on the specific situations. And that's a little bit the theme that we want to explore also for other aspects of treatment optimization. Is it perhaps a question of just being very clear in the development plans what questions will be addressed when, in a way that doesn't harm the access of innovative therapies, but that it's also not left completely in the open, so to speak, for users and patients to figure out when certain uncertainties will be addressed. Keep thinking about this. We're going to move to the next session, turn the page to reward mechanisms for post-marketing studies and generally finding incentives for optimization. We have 3 presentations. The first is by Joycelyn Stocks. Joycelyn will present Balancing Innovation and Access, Insights from AIM's Fair Pricing Model and its Current and Future Applications. Joycelyn.
Yeah, good afternoon, everybody, and thanks for having me. I will present a study we have done with the AIM's fair pricing model, which is a model that calculates what we believe is a fair price. And then I will link this later on to dose optimization or treatment optimization studies. So I'm Jocelyn, I'm a pharmacist and I work at the Belgian Christian Mutuality, and I'm also sharing the AIM working group of pharmaceuticals and medical devices, and AIM is our umbrella organization, the International Association of Mutual Benefit Societies, actually which gathers non-for-profit health insurance and mutuals from Europe and also from other parts in the world. Next slide, please. So I think we all can agree that access to affordable medicines is essential for equitable healthcare. But what we see last decade is that escalating drug prices really strain national health systems. And this is an example of this of the US, but new drug launch prices rose about 20% annually from 2008 to 2021. And so nearly half of new medicines that came on the market, they had a price above $150,000 yearly. Yeah, we can say, okay, high costs, maybe they are justified, of course, because we want to pay actually for, I think we are prepared to pay for innovation. But the HTA, the Belgian HTA organization, KCE, they did a study and they evaluated the impact of new cancer treatments. And I think I put a link to the study below my presentation, but actually they saw that there were no meaningful gains in survival, and at the start at the market time of market authorization, they saw there were still many uncertainties. And to deal with these high prices and the uncertainties, many solutions came, like we have the managed entry agreements. So I think you all know what it is. They are like secret deals between governments and pharmaceutical industry to negotiate a price. We see we don't see the real price. We don't know what the real cost is because there are discounts. It can be just a simple discount, or it can be outcome based. What we all see is that countries are starting to collaborate. We have, like in Belgium and the Netherlands, we have the Benelux project. In the south of Europe, we have Valletta. In the Nordics, we also have a lot of cooperations. And yeah, we are also thinking in the academic world about fair pricing models. And so, next slide, please. This is a short presentation of our fair price calculator. So we developed it as a transparent, cost-based alternative. We actually want a price that is affordable for health systems and patients, and rewards true therapeutic value. And it's composed of different components. And what we want is transparency. You can introduce real costs, but we know that this is not the case. So for the R&D and the production, there we are working with lump sum. So we said in the beginning, okay, we give you 250 million for the R&D costs, but if you give the real price, the real costs, it can be introduced in the model. But we made an online calculator, and then you can see you can introduce 250, 800 million, to the maximum of 2.5 billion euros. Also for the production, there is a lump sum. There is 50 euros a month when it's a chemical, 250 and 150 when it's a 250 for a biological, and it goes up when it's an orphan. But what's most important at the calculator is actually the innovation bonus. Next slide, please. In the innovation bonus, you can select what is important, what you do want to reward. And there are different options. We have this online tool, but of course, it's dynamic. You can be flexible, and you can reward what's important for you. So you can see indeed when the medicine has no alternative, it's 5%. When it's curative, it's 30%, etc. Next slide, please. So the study objective, we wanted to assess discrepancies between the calculated fair price and the actual market price of new marketed medicines. We took medicines that were marketed between 2015 and 2018, and then estimate the potential savings for healthcare systems. And we applied it across 6 European countries, Belgium, Estonia, Germany, the Netherlands, Slovenia, and Switzerland. Next slide. So we included actually 10 medicines. The selection was based on therapeutic diversity, on budget impacts and innovation. So we included medicines for cancer, for ultra-rare diseases, for chronic diseases. I'm going to the next slide. Or is there a way that I can manipulate the slides? I don't know. I can't see it directly. There is. So what we did, there's we calculated the fair price. It was based on a monthly treatment cost, and we took 800 million R&D costs. Then we actually estimated. We calculated the real cost. It's a monthly cost. Gross and net, and gross is there are the list prices, and gross and the net prices is when there is a managed entry agreement. So then we made an estimation, because we for some countries we could do this. We didn't do it for Switzerland and Slovenia, but for the other countries, participating countries, we had an estimated price. Then we made the difference between the actual market price and the fair price. Then we estimated the savings, and we calculated per medicine the savings when you have a total budget for that medicine, and then actually the reduced price calculated for calculating the budget with that reduced price, and summed it across the 10 medicines, and then we calculate the reduction rate, total savings by the total expenditure on the 10 medicines. And then this reduction rate we apply to the 2020 spending on new patented medicines. And then we extrapolate it to the national and the EU levels. Next slide. And then the key findings on what we saw that on average fair prices were more than 50% lower than the list prices. And when you take these estimations, so the real cost of the medicines into account, we saw that they were 34% lower. And you can see actually most of the time there is a huge difference between the fair price and the fair price is generally lower. But if you see, for example, for Uptravi, and you can see it in the table, there the fair price far exceeds actually the net price. And that's because, and I will talk about later, is because of the small population. But you see it for Ozempic, for example, we had like for 3 different lump sums for the R&D, we had a price of 34, 35 and 39 euros instead of a median net price of the participating country, it was 76 euros. So next slide, please. So when you go to detail in all the parameters of the model, we see actually that the R&D costs for chronic treatment, they have a low impact on price, like I said with Ozempic. And for 5 medicines, the R&D accounts for only 3 to 21% of the price, but it also has a high patient population sensitivity. So you can see that for Uptravi, you have to recoup more than 4,000 euro a month for the R&D costs. Innovation bonus, well, for 4 medicines, the bonus is only 5%, and that's because it was for a chronic for a chronic debilitating disease. And we never could give our maximum bonus because we set a maximum of 40% to any of the participating medicines. And what we see in the calculator, the production cost was a significant share of the price. For 6 medicines, this was 63 to 85% of the total price. Next slide, please. And then the savings, when we applied the fair price model, we could say that in general, the reduction rates were minus 48%, ranged from 48 to 81% on the gross expenditure, and on the net expenditure, it was minus 37 to 74% from the participating countries. Like, for example, on Belgium, we could save more than 820 million yearly, which is a lot because we have a total budget of new medicines of 1.2 billion. We had it in 2020. And then we had savings, total gross saving of 9.6 billion. And if you say net savings for the 4 countries who could give these numbers, it was 6.2 billion. And when we extrapolated, there were like savings around 27 billion for the whole of Europe. Next slide, please. So, and the savings, there were previous study done with the calculator by German health insurance and Belgium health insurance, and they had with the same numbers of savings. If you see with rare diseases, there is a small clinic. The fair price in general can be quite high in comparison to the real price, and that's actually because, and we believe that R&D costs can be lower because we have a small clinical trial population. So probably there is an adaptation needed. And what we all could see is that the innovation bonus was not always so high, and that's a bit consistent with what we found in literature. Next slide, please. And then when we actually we see that actually, is it a real model? Well, we have the calculator online. You can play with it. We have the Dutch insurance. They use it in the negotiations for a price, and currently it will be integrated into an Horizon Europe funded project. It's a certain where they actually want to make a model for HTA pricing and reimbursement, and they actually take our model as as a basis. It will be adapted, of course, and it can be used for both innovative and repurposed medicines. There were some researchers in the Netherlands who calculated a fair price for a drug that was repurposed. Next slide, please. And when it comes to the fair pricing model in the context of treatment optimization, there is, for example, a possibility of an extra innovation bonus for companies who include optimization studies in the pre-marketing phase. Also an option for post-marketing investment in treatment optimization. It can be incorporated in a model to renegotiate pricing, because of course, when you de-escalate in dose, so there will be less profit for the company, so that can be an incentive. When you see at the prices, okay, when you reduce the dose, you will actually pay less for the drug, but then the price is coming closer, what we believe is a fair price. And then, for example, by using a kind of fair price model, you can have savings, and it can be used to finance treatment optimization, for example. Next slide. So what we saw is that current prices are not justified by the underlying costs. There is a lot of margin. We want, with the price model, to enhance transparency and fairness, and we want actually to strengthen the buyer's negotiation position. We want to safeguard, and it's very important, solidarity and sustainability of European health systems. We see the calculator is very useful. It's for, you can use it in real practice, and fair price model can link with treatment optimization in pre and post-marketing phases. So next slide, please. Just to see, there were a lot of people who contributed to the study, and I'm really grateful for that. And so I came at the end of my presentation.
Thanks a lot, Jocelyn. Very clear presentation. Our next two speakers are about incentives to support post-marketing research. First, Mikhail Akimov, and then Francis Nissen. Mikhail, the floor is yours.
Thanks for having us. My name is Mike Akimov. I'm a medical oncologist and head of Boehringer Ingelheim Oncology Clinical Development. I will talk in general about incentives to support our post-marketing research, and we'll focus on one example, how we can leverage real-world data in post-marketing setting to optimize dose for targeted therapy. And Francis will focus more on challenges in cell therapy and how we can also leverage real-world data. Really briefly, when we are talking about cancer treatment optimizations from industry perspective, but not only from industry perspective, there are three key pillars what we can do better. One is increased probability of success, and unfortunately in oncology setting, probability of success is around 50–55%. It's actually lower than even in cardiovascular setting here. How we can increase it? Second very important pillar, how basically do it fast. Speed is really critical, yeah, because our patient could not wait, and we have really effective treatment, and our actually obligation to bring this treatment as fast as possible to the patient. And third, how to decrease the costs. And for all the three pillars, real-world evidence, real-world data could be crucial and critical. Again, I will mainly focus on post-approval commitment, but it's actually irrelevant. If you go to the, I will try to move to the next slide. So, yeah, sorry, probably I was a bit too fast. Anyway, just briefly about real-world, and I will focus on more post-marketing setting, but real-world data could be used in general for all these three pillars in 4 different settings. One is for early trials, for proof of concept trials. We will not talk today, but basically external controls could help. I think some of our colleagues will talk later today about pragmatic or synthetic control arm in registration trial, which we for sure can do better in oncology. And we will focus more about post-marketing settings, and there are 2 different settings. It's really how to generate more safety data, follow-up patients, randomized control trials, and observational trial, and I will talk a bit about the afatinib case later today. A few words about incentives. Sorry. Either it's not working or... Okay, let's move next. Just a few words about general incentives. And first I will talk again from industry perspective about regulatory incentives. Here, probably the first one is extended market exclusivity, which for pharma companies is quite attractive. And especially we use this for orphan disease or pediatric studies. And we all know that 6 months additional exclusivity actually boost a lot in pediatric setting. And a lot of pharma companies conducted pediatric trials in this setting. Accelerated and conditional approval, and we have a lot of examples where we basically use surrogate endpoints and real-world evidence for accelerated approval, but we need to conduct post-marketing or confirmatory trials. And third one is the label expansion and opportunities to expand label, and I think real-world data can play a great role here. Second pillar is scientific and strategic incentives. Please, next slide. Again, very important to improve safety risk, and we all conducting, and I will talk a bit about safety observational trials. I just put here example, when I started my career, I focused on first generation EGFR TKI, and we did, I think, a good job to do a post-marketing evaluation of IOD events for first generation TKI, and we start to understand better this type of complication, and actually it's helped to second or third generations. Real evidence collections which support label claims, pricing negotiation, and new formulations. And I think quite important also to strengthen stakeholder relationships, and we are working also with patient organization on this, and I think here also post-approval data collection very critical. 2 next pillars, please, next slide. Policy and system level incentives, regulatory incentives, I think frameworks and EMA and the FDA, I think, and other authorities encourage us to design and do more post-marketing trials. I think it's quite important also for pharma because it's improved corporate reputation and influence policy development. Data access and infrastructure support, I think it's also quite important. And last but not least, the last slide, is to talk about financial and market incentives. And I think, as I already mentioned, reimbursement is quite critical, and we use real-world data, good example in Germany, to encourage post-launch evidence and generation, and to increase robustness. For example, if we use PFS as a primary endpoint and we collect more survival data in real-world collection. And of course, competitive differentiation real-world data could be used. Now, a few words about a relatively old example on afatinib, but I think it's a good example how basically company took a, I would say, tough decision to look on real-world data and to see how we can optimize the dose. Next slide. We know that in our clinical trials, we select the patient. These patients are not always reflect the real world. And in post-approval setting, we see the difference in safety and efficacy. This is, I think, quite important to look on real-world experience, to collect more data, to look on a patient with a poor prognosis, and to understand better the safety profile and dose we used even in post-approval setting. Next slide. Next one. Yeah, this one. A few years ago, BI, after initial approval of afatinib, decided to collect and conduct observational real-world data trial. It's a global trial in 29 sites with 13 countries with 228 patients involved. And the idea was to look on adverse reaction and to see if our selected dose, and approved dose was 40 milligram, is right for all patients, or we can further optimize this. Next slide. Again, this trial was conducted, as I mentioned, the primary endpoint to look on severity of adverse reactions, but also to look on time to failure and time to progression. Next slide. This trial was conducted in first-line EGFR mutant setting, and as you can see, Based on this observational style, we find that some patients, regardless of start dose below approved 40 milligram with 30 milligram, drive exactly the same benefits. The time to failure and time to progression were identical for patients who started with 40 milligram and 30 milligram, which actually helped a lot to guide and use dafatinib, for example, for the patient with a high risk of grade 3 events or diarrhea, elderly patient or patient with a low risk, and basically in general practice to use a lower dose. My last slide, I think, for today is, again, I just want to encourage also my colleagues in industry to conduct more this observational trial, to look on data in a real-world setting and really better understand what can be used and how we can optimize the treatment, and actually not scared to optimize the dose even in the later settings. Now, Francis, your turn.
Thank you very much, Mike. So my name is Francis. I'm with Kite Gilead, and so I'm working mostly on post-marketing studies in cell therapy. So I really appreciate the opportunity to share our perspective today. Next slide, please. Yeah, so in terms of I'll talk about various incentives for post-marketing research, and also including how the field can leverage the growing knowledge so as more real-world evidence becomes available. So in general, so of course, post-marketing studies, they can either be interventional, I mean, so-called phase 4 clinical trials, or non-interventional, which are the observational studies. And so there's a wide variety of aims, objectives, and especially for the observational studies, many different study designs. Next slide, please. So if you zoom out a bit, so of course, from industry perspective, there's a lot of different incentives for us to do post-marketing research, and so of course one of the key stakeholders is of course the regulator, because while regulatory approval is a major milestone, it's not the endpoint. In real-world data and a continued evaluation remain essential. But then, of course, also quite a bit of the post-marketing research, as we also heard from Mike and Jocelyn earlier, is also for the payers and for the reimbursement decisions. And for example, the value-based pricing is often tied to post-marketing evidence generation. And then finally, of course, there's also the clinical decisions, so the physicians and medical societies, where gaining additional insights of study questions that have not been addressed by the registrational trials can still be addressed by additional post-marketing research. So next slide, please. So in addition to the exclusivity that Mike already mentioned, there's of course the conditional marketing authorization, which is a big incentive for the post-marketing research. Also, for example, the PRIME scheme with enhanced support and early dialogue for promising medicines facilitates and shapes the research agenda, which may include post-approval studies. And then, of course, there's all the mandated post-authorization studies, so the post-authorization safety and efficacy studies. Next slide, please. So now I'll go a bit deeper into one specific case study, which is the long-term follow-up for cell and gene therapy. So based on the 2018 guidelines on the safety and efficacy follow-up and risk management on advanced therapy medicinal products, there is currently a 15-year recommendation for 15 years of follow-up, so both for clinical trials and also for the post-approval safety efficacy studies. And this mostly based on a theoretical risk for insertional oncogenesis. And so this was also a precautionary measure when little real-world data was really available for CAR-Ts and for gene therapy. So it's relevant for the current discussion as the 15-year follow-up does create a barrier for additional post-marketing research in cell therapy. Next slide, please. So currently there's more than 32,000 patients worldwide that have been treated with CAR T-cell therapies since 2017. There's been 7 CAR T-cell therapies approved. And there's almost 15 years of data that has been accumulated since the start of the cancer CAR T clinical trials. Next slide, please. And so to evaluate actually the evidence of where we currently are with what evidence is available on the longer-term follow-up for CAR T-cell therapy. So we brought together 21 authors for a white paper, so including voices from patients, academia, industry, and the US National Cancer Institute. And so the process included just looking at the compilation of long-term follow-up challenges from the patients and from the providers. Assess the global safety databases with also a systematic literature review, and then also identify new technological capabilities. Next slide, please. And so what we saw is here's on the left. So this is what we saw is after 5 years, so the reports within the post-marketing studies are quite rare, so approaching zero. So then after the 5 years, keeping the study open for another 10 years might not yield really meaningful safety signal when there's no additional reports coming in. And so on the right, you can see a literature review from 13 published studies, where we see that the second primary malignancies, which is the main reason for the long-term follow-up, are similar to the background rates in patients that have received other treatments. So CAR T-cell malignancies are very rare, and a causal link to CAR Ts has not been established. Next slide, please. And what we also see is when key adverse events do occur, they tend to occur early. So, for example, from the FDA's data, it showed T-cell malignancies occurred generally within the first 2 years. And so the published studies showed most malignancies occurred within the first 5 years. Now, one of the centers that has the longest experience with CAR T-cell therapies, so the UPenn, University of Pennsylvania, they have over 25 years and 783 CAR T patients in oncology and autoimmune patients, they're spanning HIV and pediatric as well. They found only one case occurring after the 5th year, which was a thyroid cancer case at year 14 in a patient with previous history of radiation to that area. Next slide, please. So with that said, so the overall recommendations of the white paper were to shorten the long-term follow-up requirement for CAR T-cell therapy, but then of course that can be to extend based on what is seen within the first 3 to 5 years of follow-up. Then also to streamline the adverse event data collection using electronic health records, which could enable comprehensive safety data collection even beyond the long-term follow-up period and decrease the burden on healthcare providers and patients. And finally, focus data collection on events that are clinically meaningful and mechanistically relevant. Next slide, please. So that's all from my side. Thank you very much for the opportunity.
Thank you very much. Our next speaker is Zahar van Waalwijk van Doorn-Khosrowani. She will speak about innovative reward mechanisms. Zahar.
Hello, everyone. There's continuous discussion on dose optimization, and I'll go further with your study, Francis. Try to give another example from the Netherlands. I'm associate professor in medical oncology with focus on accessibility of medicines, and I work with one of the payers in the Netherlands. And on behalf of the payer says that I'm part of the national committee for the evaluation of medicine and molecular diagnostics committee that decides on the reimbursements of medicine. There was just a very recent publication by MEB, our national competent authority, interviewing different regulators and healthcare providers, just published, showing that healthcare providers continuously struggle in finding the right doses for patients and using drugs off-label as there is no evidence. And as payers, we are also often asked to support those optimization studies, and we try to be supportive. Recently, we published a guidance on what can be reimbursed within a trial to try to reduce the cost of such trials for the sponsors of the trials. So basically, in the Netherlands, you can get the regular care reimbursed within clinical trials, but for this type of studies, you can also get the experimental arm of the trial reimbursed. The paper that you see on the screen is the one we published a few months ago, different stakeholders, and we tried to see what are different types of dose optimization studies. Of course, you can go for less drugs or stay with the same amount of drug, or find different dose schedules, or go for more drug. And that can be also by adding another drug to your therapy. And the main outcome should be better cancer therapies, so better overall survival, better quality of life, but also reducing use of resources and budget for the society. We also provide a guide for how to prioritize such dose optimization studies. Of course, the highest priority should be given to the therapies, dose optimization studies that improve efficacy and reduce side effects and improve quality of life of patients. But I would argue that using less drugs, reducing the burden of the treatment for the patient, but also for the society in terms of financial toxicity, is also something that increases healthcare, maybe not for those specific patients, but as you free up resources for other patients, then you're also improving health for others. If I may go to the next slide, please. The example that I would like to discuss with you today is adjuvant therapy of in melanoma stage 3 or resected stage 4. And about 7 years ago, 2 different schemes were authorized by the EMA based on Keynote 0454 and Checkmate 238. And both of them use 1 year of adjuvant therapy of immune checkpoint inhibitors. If I may go to the next slide. So this is our claims data, number of patients that received one of these 2 schemes between 2020 and 2025. And as you see that there is a decline, continuous decline in the number of patients receiving these therapies, and that the reason for that is the lack of trust in these therapies. There is no overall survival advantage. There is a high burden for patients to receive these therapies, also in terms of long-term toxicities, but there is also a financial toxicity for the society. So the numbers are reducing, and if I may go to the next slide. The good thing that these 2 companies, MSD and BMS, did was to invest in dose optimization studies. So we have the SWOG trial. It's an investigator-initiated trial sponsored by MSD, and they divided the dosing of adjuvant therapy in neoadjuvant, 3 doses of neoadjuvant and 15 doses of adjuvant therapy. And the other trial, if I may go to the next slide, is NADINO trial, also another investigator-initiated trials from the Netherlands. And it is sponsored by, it is financed, the sponsor is Dutch NKI, but it was financed by BMS. And in this trial, they chose for actually an escalation in neoadjuvant setting, because ipilimumab is added to nivolumab for 2 cycles. But in this way, the trial achieves more major pathological responses. About 59% of patients reach major pathological response, and then no added adjuvant therapy is needed. Only the patients who didn't achieve response, then they go on with 12 cycles of adjuvant nivolumab. So this is clearly those de-escalation studies, except for the first part, the neoadjuvant setting, which is dose escalation. What you see is the updated results of the SWOG trial and NADINO trial that was presented a few weeks ago at ESMO. And as you see, both trials show long-term advantage versus the authorized indication in red. So the NADINO trial, the upper figure, shows 21% advantage in event-free survival, and the SWOG trial, 15% versus adjuvant pembro. And both trials are significantly positive. Now I show you again the claim data from the Netherlands in 2024, and 2025, the data up until now. And as you see, so this is actually the first half of 2025. We still need to update the data at the end of the year. But as you see that between these 2 years, the adjuvant therapy use is even more reduced. As I said, lack of trust in those authorized indication, as no survival benefit was demonstrated. And then for NADINA and SWOG, the lighter bars here, you see that these optimized regimens are being used more often. And as you see, NADINA trial is being used more now even than the SWOG trial. I should say that if you look at the data of 2024, the SWOG trial, the results were published earlier, and there was already reimbursement in the beginning of 2024 for the SWOG trial, while NADINA data were published later, and then the reimbursement were in place at the end of 2024. But probably looking at here, NADINA would be the regimen of choice in the Netherlands, as it leads to less use of resources. The summary. So this case study shows that post-marketing dose optimization studies could lead to competitive advantage for the company, because otherwise somewhere the healthcare providers would just stop using those adjuvant therapies. But NADINA and SWOG helps reaching actually better trial results, and resource-saving schemes are encouraged by hospitals and payers. So those would be schemes of choice, and also improved efficacy and safety profiles result in stronger clinical trust in those regimens, and that this is both better for patients and society. Thank you.
Thanks a lot, Zahar. Very clear. Colleagues, we have a few minutes for a quick comment or a question. What are you thinking about it? I think we had 3 fantastic presentations again. Jocelyn, I really liked your model of the innovation bonus. I wonder if it could be turned around to incentivize treatment optimization. I think in your last slide, you said it could. And Mike and Francis, you of course look at real-world evidence as also another source of evidence to answer some of these questions. And I've heard at the ISPOR that the EMA is a world leader in real-world evidence. So we are certainly listening to you. And of course, the studies that we can impose are diligently conducted. The question is, the study that we cannot impose, are they conducted? And Zahar, you gave an excellent example where collaboration is possible. So colleagues, if there are no questions, then let's go straight to the next session, staying on the theme of collaborations and partnerships, industry's role in supporting optimization post-approval. First speakers are industry support for pragmatic trials and real-world evidence studies. First speakers are Nafsika Kronidou and Liz Poole. Nafsika, the floor is yours.
Good afternoon, everyone. My name is Nafsika Kronidou. I work for Roche, but today's presentation I will be focusing on work that has been done by an FBI working group. Okay, so for today's talk, I'll tell you a little bit about why I think that we should be revisiting the concept of pragmatic trials. How do we actually design, how we could design trials with pragmatic elements, and we'll also share 2 case studies as a way to conceptualize the concept and understand the opportunities and the barriers. And then I will finish with some final reflections and potential future areas of what we could be working on. So the concept of pragmatic trials is actually not new. Some of the publications I've read date back to the mid-60s. But in fact, there are very few trials with pragmatic elements which actually led to regulatory decisions. And so I'd like to, today's talk and follow-up discussions, I'd like to explore this concept and why we haven't been actually using more pragmatic trials for regulatory decision-making. On the next slide. So let's start with the problem statement. I think most of you would agree that the current traditional clinical trials are generally have increased in complexity. We collect much more data, and that it translates to a lot more burden to the patients. If we have to, for example, we have to bring patients on a regular time points to collect that data, it is adding a lot of more burden to them. In addition, if you look at the number of patients who are participating in clinical trials, the latest publication I saw was that only approximately 7% of patients with oncology actually participate in clinical trials. And if you also think about the actual data that come from traditional clinical trials, they don't always reflect the evidence which reflective of clinical practice and what physicians would actually need to treat the patients in clinical practice. There is, and then there is the final topic about mindset and perceptions. I have a hypothesis that I'd like to explore with all of you, that most of us, scientists, think that more data is better. And that also regulators would never accept more streamlined data collection. So let's explore and dig into this topic a little bit further. Next slide, please. So on a positive side, and if we think about what are the opportunities that randomized pragmatic trials could bring, I feel that they could be much more patient-centric because they would reduce the burden on patients if we have less visits, if they're there, and we do more fit-for-purpose data collection. By that I mean data which is required to answer a benefit-risk question. We could be also generating more patient-relevant outcomes beyond mortality, of course, such as patient-reported outcomes and real-world endpoints, with the intention of those evidence and those data to inform benefit-risk decisions. And I talked earlier already that if we have more pragmatic trials, then hopefully that reduced burden would encourage patients who actually want to participate in clinical trials to actually do that. And let's not forget all the technology which is out there, right? Right now, the evolving technologies. My colleagues before talked about the evolution of real-world data, and think about the investment that we are doing in the healthcare space right now in terms of generating higher quality real-world data. And therefore, the ability to leverage electronic healthcare records and data from digital health technologies, and that longitudinal objective data collection that could be collected through digital health technologies. And I talked already earlier about the external validity, that if we generate evidence closer to clinical practice, hopefully the generalizability of that data would also increase as compared to the traditional clinical trials. Now, there's a little bit of a messiness in terms of terminology. As you can see on this slide, there are tons of different definitions out there. But at FPA, we decided to focus on the definition of trials with pragmatic elements. And the reason we chose this definition as opposed to everything else is because this definition outlines and shows that it's not an all or nothing. In other words, the data, the evidence, the scientific question will inform us what pragmatic elements we should be using for our trials. Next slide, please. Perhaps you've seen this tool before. It's called the PRECISE-2 tool. And we have found it incredibly useful, one, to explain to people what we mean by pragmatic elements. And also, it is a tool to actually help people to design clinical trials. An important element, and so let's take with the PRECISE-2, it talks about 9 dimensions of pragmatism, and within each dimension, there is a spectrum of pragmatism. So let's take eligibility criteria. As opposed to the traditional clinical trial, where we usually have very highly controlled conditions, in a pragmatic trial, and if we have the relevant evidence, we may actually start increasing broadening the eligibility criteria so that we actually, our trial could eventually reflect more the patients in clinical practice. This is a dimension of what I've said before. So in a traditional randomized clinical trial, we usually conduct those studies under highly controlled conditions. But at the time of the approval of a molecule, we will be gaining more and more experience with the molecule, and therefore there will be more data following the authorization. And that data could actually help us understand where we could introduce pragmatic evidence, that understanding on the mechanism of action, the broader understanding of the safety of a molecule could give us confidence, and hopefully also regulators, that we could introduce more flexibility and more pragmatic elements. Now, at the time of an approval, the first initial approval, and despite all our efforts during the development of the product, there might be additional evidence gaps that we may need to actually generate post-authorization. And that could be new indications. We may also want to investigate different patient groups, for example, the elderly, pediatrics. There might be comorbidities that we were not able to assess in the initial phase development of the molecule, et cetera, et cetera. And that evidence from randomized pragmatic trials could be used to inform treatment guidelines, but it could also be used for regulatory decisions, which is of particular relevance for us in the industry. And so now I will talk about two case studies in oncology. These are hypothetical case studies, and I would like to use those as a way to conceptualize the opportunities and the barriers that we are facing. And both hypothetical case studies are on treatment optimization, of course, consistent with the theme of today's meeting. Next slide. In this specific case study, there is, following that initial marketing authorization, we recognize that there are some evidence gaps that we unfortunately were not able to address initially during the initial 2 pivotal studies. And this is for elderly patients. And our assumption, and in the future, hopefully with more interactions with patients, is that, in fact, this specific patient population would probably value even reduced visits and data collection. So let's say we select for this patient population, and based on the available evidence, select more selective safety data collection, and by that could take different shapes and form, but in this particular case, we are making the assumption it would be reduced frequencies of assessments and focus on severe adverse events. We would also want to collect, in this specific case, more tolerability and patient-reported outcomes, and hope that we can use that data in the benefit-risk assessment and in, of course, also including that information in the patient information so that it could inform patients about the potential toxicity of the molecule. The next case study, it's a case study of it's another sort of treatment optimization. The standard of care in this case study is chemotherapy plus cancer immunotherapy induction followed by maintenance with cancer immunotherapy only. We have compelling evidence from a phase 2 that, in fact, that by adding an additional agent in the maintenance phase could really improve the outcomes. And so we want to do a randomized pragmatic trial to confirm the evidence, the data from the second. The phase 2. And in this specific case study, we want to leverage electronic healthcare data and death registries to capture mortality outcomes. Here I've already outlined all the opportunities with introducing pragmatic elements, whether expanded eligibility, using endpoints which are very more relevant for patients, streamlined data collection, registry-based trials, and electronic healthcare records. But the question at hand, and I think that's what we all need to resolve, or what are the actual or perceived risks? Because anyone who would like to engage and actually conduct a trial with pragmatic trials would want some confidence informed by data and or other data or other information, and confidence also that regulators would accept that. That if they conduct the study, it would lead to robust data that can be used for regulatory decisions, and that it will also be, the study design will also be acceptable by regulators. So I think I want to spend more time about this actual or perceived risk, because, I think I touched on most of them. But let's go to the final slide, which was also informed by a recent FPA survey in terms of our experience by industry on conducting trials with pragmatic elements. And the conclusions from this survey point to 3 major elements. The first one, that we need to build broader awareness of trials with pragmatic elements and the benefits of such trials. We need to all build expertise in designing such trials, and we need to build confidence that such evidence can actually inform regulatory decisions. And in order to do that, what we believe is important is that there are more case studies where we can actually co-design the people in the healthcare system, whether it is industry, academia, patients, real-world data custodians. We can together work on potentially case studies and find possible solutions. There might be methodological elements as well through this co-design of trials with pragmatic elements. We may decide that we need to develop more methods, and hopefully we'll through these learnings from the case studies and communication, whether it's through publications, through conferences, etc., we can build this collective experience and build trust and confidence that we can actually execute such trials. We would like regulators to be there with us so that we can, to help us in the design of such trials, and so that we can have more confidence. And of course, we will also need global alignment in terms of some of these principles. And in Europe, where we have a relatively complex ecosystem, it's going to be also important that EU member states are also on board along in this co-design and learning environment, so that when we submit our clinical trial applications and also when we submit our data for marketing authorization, there is some alignment in terms of the approach that we've taken, and therefore confidence that we can actually execute such trials. And then my final point is around, I'm going to go back to this mindset shift that perhaps there is belief in our societies, a scientific society, that more data is better. And in fact, all our guidelines talk about risk proportionality principles, quality by design. The principles are all there. They're wonderful principles. But the question is, how can we translate those principles and actually show to society and regulators and to within our industry colleagues that, in fact, maybe more data is not always necessary. Maybe we can we can come to benefit risk conclusions with less data. And the next final slide, sorry, it's an ecosystem, right? And we'll take a village, and it's going to be all the people involved in this ecosystem, in this healthcare ecosystem, need to co-design, and we need to learn to actually design trials with pragmatic trials for the best interest of patients. Thank you.
Thank you. And our next speaker, Denis Lacombe, will talk about collaborative models with academia and research institutions.
Okay, thank you, Francesco. So I'm going to do a little bit something different than usually I do in the Cancer Medicine Forum. I'm not going to too much to speak about treatment optimization, but about potential collaborative model. Can I have the next one, please? So just to put a little bit some context of what I'm going to say, because there can be very different models of collaboration between industry and the academic sector. But of course, the objective when we created the Cancer Medicine Forum, the objective was to look at the potential to uptake the work of the academic sector in regulatory decision making. So here I'm going, of course, to refer to large international clinical trials which are potentially practice changing and which could eventually change the label, because that's where we put the ambition if we really want to address patient-centric questions. Sorry, can I have the next one? So we tried a little bit to analyze, and maybe there are other reasons it's not necessarily exhaustive, why industry would collaborate with the ORTC, and we maintain a constant dialogue with industry on the evolution of the needs. So it's access to specialized network. Sometimes it's access to methodological expertise. And for instance, the ORTC has published and developed quite a lot in the field of pragmatic clinical trial, pragmatic elements, how we can do that. Often it's to achieve more beyond the primary indication, and possibly we can put there a little bit treatment optimization, although I must say that in the ORTC agenda today, our treatment optimization agenda is mostly a purely academic agenda. We are often also approached for IITs that cannot be handled by CRO, and also, of course, is to keep a scientific dialogue open with learned society. And of course, for us, is to access to a pipeline of new agents, contribute to therapeutic progress, address the gaps in scientific strategy, and that's where we position our question for treatment optimization. And as part of that, of course, and it was mentioned earlier, specific interest for patient population which are not necessarily addressed in the development plan, rare cancer or older patients, if we remain in the optimization field and for the same drug and the same indication. So while I prepared this presentation, I said, are there clinical situations which are more likely to support partnership and where potentially we can discuss in the future the evolution of the partnership between the commercial and the academic sector. So there are many. It can be extension of label to new tumor types not primarily targeted by industry. It can be extension to earlier line of treatments in the same indication, and we have seen some example earlier this afternoon. Again, access to specific network for recruitment, which are otherwise not easily attainable, especially, for instance, in the ORTC, we have had good success with partnership with industry for access to the neuro-oncology network or the sarcoma network. Again, access to specific patient population, optimization in patient subgroups, like further biomarker research, or also when there is a need for cross-cutting expertise. And for instance, an example that is current is radioligand multi-tumor, where you need disease and imaging expertise combined. Then the next question I try to ask myself is, are there types of clinical trials which are more likely to support a partnership? So certainly complex clinical trials which include a strong quality of life PRO component with applied quality of life research, and EORTC as a good, excellent basis for methodological research in this field. Also access to patients who for instance, for trial within cohort, when we have a registry in place, pragmatic trial, and again we have published in that respect, and of course multidisciplinary clinical trial, especially for perioperative trial or combination with radiotherapy. So there are a number of areas that we can discuss and that we can optimize. I think that for this also, and EORTC is not the only one. If we think that we want to partner again with the potentially to have an impact at the regulatory level and the access level, because we tend to place treatment optimization more in the late stage disease, is that also the academic sector has to make an effort for the partnership and in the quality of the data set we can deliver. So we are constantly evolving in that respect in terms of, for instance, all the expertise we can have at the EORTC in terms of developing a regulatory compliant clinical trial protocol, adapting to the evolving guideline. We are talking about ICH 6 revision 3, all this type of thing to be on the top of the changes, while ensuring, of course, peer review methodology. And I think what is very important also is to have a very flexible environment. And at the EORTC, we try, and we are in the context of our policy work at the European level, connected to HTA body, to payer, and the Cancer Medicine Forum has been quite instrumental, of course, in further developing what we can do between the academic sector and what I would say, the downstream players, so HTA and payers. Although now the HTA with the joint assessment are really at the beginning. Can I have the next one? Something also that has been important in the evolution of the network. Again, keep in mind always this intention to have regulatory compliant data set in time is to, of course, implement solution with professional project management to meet the standard, and of course to access to patients, not only for recruitment but also to develop the protocol. And I will give an example. And of course all the infrastructure and the know-how how to organize this sometimes complex clinical trial. Can I have the next one, which actually is very important because that's the title of the presentation, is the different models of collaboration. I would like to drive you relatively rapidly through this 3-column table with the model of the investigator-initiated trial, which I think is not really for discussion today, but more between the collaborative and the registration model. And we can go through the different rows first, and I will focus mostly on the two right side columns, where basically we can discuss in the collaborative model or in the registration model who is the sponsor, and of course who has the control of the data, if I can say so. I use the word ownership, although I don't like ownership, but I think it speaks to it speaks to people. I think in a new RTC-like environment, we like to keep control of the protocol, at least the synopsis, the final analysis, and the publication, and access to the final study report is, of course, in the two model, as well as access to the safety data, because of course you need to report as an industry. What changes, of course, between the collaborative and the registration model is that in the collaborative there is a non-exclusive license to access the study data after the final study report, but there is, I would say, a possibility for an option for an exclusive license under a separate license agreement that we can discuss. In the registration trial, of course, this is the case up front, and of course there are differences between the resulting budget. So we think that we are open for discussion what is the best model, especially for treatment optimization, and potentially the collaborative model is an interesting model. Again, to continue developing knowledge about a certain product, with the possibility, of course, to impact on its on access. But the two models could be suitable. Can I have the next one? I think what we are trying to achieve through this work, through this partnership, is that, of course, industry has the know-how how to develop innovation and bring innovation to the market. But again, there is room for optimization and how to use this therapeutic intervention optimally. And I think here there is a gap, and we can contribute in partnerships in filling this gap, especially at the time where it was said earlier also that of course registration of a new marketing authorization of a new drug is important, but the challenges potentially remain in access and how we can have better information on how to use therapeutic innovation. So if you can push through the different arrows, but basically if we have to rethink can you push 1, 2, 3? Yes. Thank you. If we have to rethinking the partnership between the commercial and the independent sector, we can help optimize the role of stakeholder alongside the process of development and access. Maybe there are things indeed where industry is best positioned to do, but maybe there are things where the academic sector is best positioned, and how we can optimize that. I think we can build on opportunities for treatment optimization. You've seen the question on treatment optimization, de-escalation have been really flourishing. At least at the EORTC, we have several trials on the table for optimization. I think you guys at industry, you may not have completely exploited the power of independent research and what we can and what can be achieved. And I think if we rebuild that, it would be very important to rebuild with all the stakeholders, because then we can really re-engineer the process, especially for this population which are not included necessarily in regulatory trial. Again, the objective is not to delay access to innovation, but it's to make sure we have a good understanding how to use this innovation. And I think it was said better by others before me, to contribute to a sustainable economic model for cancer research, development, and access, because indeed we cannot deny that as a society we have major economic challenges to take. So I just would like to show an example of a clinical trial which is currently ongoing. So it would fit in the first column I've been showing you. It's really an investigator-initiated trial. At this time, it's not a collaborative model. It's not a registration model. It's addressing androgen receptor pathway inhibitors for men who have a certain control of their disease. And the de-escalation is basically randomizing between the continued treatment, which is the register schedule, versus an intermittent treatment. And of course, these trials have raised challenges. It's a non-inferiority trial, and we design it with patients. And this is maybe something that, as a network of investigators, we have more agility to do. We really discuss all these aspects of this trial with the patients upfront. Why? Because again, what is the acceptability of a de-escalation, and also with the investigator, because what we didn't want to be exposed to is trials which is not going to recruit. And if you prepare well, you can't see, because you see a little bit of the start of the recruitment, because we just opened this trial 3 months ago. We are still in the process of opening the different institutions, and in 3 months we are really on the curve with today, I believe, a little bit more than 45, but 50 patients. So this translates that the fact that you need to develop a know-how, how to do this trial, how to discuss with patients, the buy-in, and also with the investigator. And this is something, it's a learning curve, and I think there are learnings indeed that we can share, because these trials are not necessarily a given. For your information, this trial is supported by a grant of the Cancer Mission of the European Commission. Can I have the next one? I just want to say that this is a very pragmatic trial in line of the previous presentation. I think we have 4 eligibility criteria. We collect minimum safety data, because we know this agent. They are used in everyday practice. And can you push another one once? And of course, what is very important is to improve the quality of life and reduce toxicity of cancer patients. Can I have the next one? And I believe it's the last one. Now that you have heard a little bit where I'm coming from, why EORTC has really stimulated the creation of the CMF, and I'm really thankful to the EMA and to Francesco for following us, is because we were confronted with our network of investigators of many questions which were not addressed at the time of marketing authorization, and because of different factors, the regulatory environment, the cost of price, the cost of drugs, and so on, it's been extremely complicated more and more to do this optimization trial, which we have done for decades. The environment is changing, and we have to address them differently. So we have been confronted with all these questions about dose, combinations, sequence, schedule, patient subgroup, specific population like elderly again. We wanted really to discuss the regulatory acceptability of this trial. We have gained a good understanding, because indeed at the beginning we were not sure about how this trial would position. And again, something that is very important is that optimization is not something for at the end of the treatment. It's a continuum, and maybe we have to discuss how we re-engineer a little bit this different question about optimization, and indeed being with pragmatic elements, what can we start as early as possible until potentially the full deployment of all the pragmatic elements, like in our prostate clinical trial. And I think there are challenges with this trial. For instance, they are not necessarily easy to put into our system. The clinical trial regulation is not necessarily completely fit to accept this trial. So I think we really need to work with all the stakeholders to address the cancer policies in general, and how we are going to continue to bring therapeutic progress to cancer patients. So I think you see it's a presentation that is combined kind of not only of operational model, but also about what we can do together at the policy level. I thank you for your attention.
Thank you, Denis. Colleagues, we have a little bit of time for some comments or questions. Please raise your hand in the system. In the meantime, I really thank you both for two excellent presentations. I think a lot that I heard is about perceptions and sometimes debunking myths, because on one side there is this perception that independent trials might be academic in objectives. On the contrary, these pragmatic trials address very relevant questions in a context that makes the results even more relevant for practically all decision makers. So that's something that we need to change. And also, Nafsika, no matter how often and how clearly we write it in guidelines, like take the annex to E6, take the actual E6 that simplifications in trials, when they make sense, are not only possible but desirable, there is this perception that the regulator doesn't stick to it, that these statements are merely theoretical. So I wonder, when we come back to the theme of this, whether really a lot is not up to understanding each other better and gaining more confidence and relaxing a little bit all of these anxieties that at some point some regulator somewhere might say, oh no, you haven't collected this concomitant medication, therefore I have a major issue. And I think we need to really understand each other better. And perhaps as regulator, definitely we should take our share of responsibility. Perhaps we're not communicating how these pragmatic elements are not only acceptable, but often desirable. We have Karine Cohen, who raised her hand. Karine?
Yeah, hello, thank you. I wanted to make a point. Actually, it's everolimus. It's the same drug that is used across different indications, both in cancer and in patients with transplanted organs since over 20 years. And there's a misalignment on the safety measures across indications, which to me raises major concerns. So in transplant patients, surgeons established that you should not stop treatment because, of course, you have a risk of rejection of the organ and you should not trigger adverse events. And therefore, they implemented the monitoring of the blood concentrations because we know that there's individual variability in the metabolization of a drug as well as during treatment in case of complications or liver issues or whatsoever. So they monitor the blood concentrations to keep it under a certain safety threshold over time. While the oncologists, they decided like 20 years ago, what was like a maximum, approach. You described that you should start with a maximum dosage and until you trigger adverse events, and then you either withdraw the drug or you decrease the dosage. And because they established that without looking at the blood concentrations, there are many people who you don't even know if they have a response to the treatment because they stop treatment after 3 weeks because they go to a hospital or they die, because of adverse events. Other people are underdosed, and the oncologists refuse to do this monitoring of the blood concentrations. So I wonder how you could address that, please.
Yeah. Thank you, Karine. And I don't want here to start a discussion on individual situation, but as you mentioned, dose optimization is clearly in scope of treatment optimization activities, and we should clearly learn from the many questions that are still open. Colleagues, anything else on the sessions we've heard about, or shall we break early for coffee? I see people nodding in the room, so I guess we'll go for the second option. Thank you all for your presentations. Thank you for the questions. Welcome to session 4, data sharing and tissue banks moving forward. Professor Jonas Berg, you will tell us about opportunities in sharing molecular clinical data. The floor is yours.
Thank you very much, Francesco. It's a pleasure and honor to be here to discuss with this distinguished auditorium. I will discuss opportunities in sharing molecular clinical trial data shortly. I'm professor of oncology from the Karolinska Institute since 25 years and in Oxford since some 8 years visiting professor. And I may know a little bit about sophisticated advanced science because I've been member of the Nobel Assembly and working for the Nobel Committee, as you know, being involved in delivering the Nobel Prize in medicine or physiology. So I know a little bit about this complicated topic we are discussing today. And there is no question that innovation and early development by pharma industry have been the cornerstone for access to novel, many times innovative cancer medicine during the last decades. And they have been using the better understanding at the molecular level for rational drug designs, and that has been resulting in better drugs, which has improved the outcomes at best, and also at best with decreased breast cancer reduced breast cancer cancer mortality. And I've been working with academic group and the pharma industry in various types of studies for some 30 years. And I know a little bit about how EMA works because I was a member in the scientific advisory group from 2004 until 21-22, and the last 5 years acting head for the oncology hematology. And I do have some also experience from being advisor to FDA. And I've been involved in different intergroup activity and specific research group activities, as well as interaction, obviously, with the EMA and FDA and some US intergroup. And over the decades, it has been the tradition that investigator-initiated clinical studies have received support from the pharma industry or been sponsored by the pharma industry. But as you will see, those type of, next slide please, these type of collaboration have diminished over the years. And that is, for me, a serious concern. And it's very obvious that the industry seems less interested in running collaborative studies of that previous type. Some companies see this as a positive development. Others are more concerned, or the translational science may risk to be more superficial, investigating and understanding mechanism for effects, side effects, and identify clinically relevant subgroup may not be given priority. EMA has a very good wordings in their documents regarding conditional marketing authorization, and that sometimes it would be a benefit to have approval, despite that all jigsaw parts of the puzzles are not in place. But then we come to how ambitious the applications are. I know a little bit about this because I've been involved in the approval of 100 new drugs indication when I worked for EMA. So it's either so the application just passed the bar or with some margin, which include trying to understand and better explore and understand the tumor biology, which may potentially enhance the benefit-risk balance. And then the question is, how should additional data be obtained? Patients, of course, they have the ownership of their blood, their tissues, their cancers. These individuals, I personally think it would be great if they can be further involved in the approval activities. And primary approval for the first indication sometimes will be a conditional type. That means that you don't have all the information. And I can give you an example. Immune therapy to triple-negative breast cancer increases the PCR rate, and it also improves the outcome. but there are other things you would like to know, and PCR is not the only thing which is a prognosticator. It should, of course, be important to discuss how extensive should the initial approval process be. Is additional therapies post a partial complete response needed? Because nowadays, if you receive a complete remission, based on the studies, you should continue with the therapy for many months, despite that the survival curves as demonstrated by Patricia Cortisar in 2014 indicate that the vast majority of patients with complete remission are doing very well. How do companies work in order to have the full approval? EMA and the market authorization holder / the company are the only involved players. Who else should be involved? Patients have the ownership of their blood tissue cancer. They should, of course, be involved. Academia and potentially also payers should be involved sometimes during this process. MHAs are very good to fulfill the requirement by EMA and FDA based on the present rules, guidelines, legislation. Advanced and frontline strategy with the most advanced multi-omics may not be used, thereby potentially missing options to even better analyze the benefit-risk balance. How to understand inter-patient difference in the anti-cancer effects and side effect profiles, albeit that the overall benefit-risk balance may be positive. The heterogeneities are not captured, characterized, and studied. Liquid biopsies are frequently used today, but there are no studies who really have compared the usefulness of conventional tumor biopsies containing both tumor stroma and the cancer cells. These two compartments are together highly important, but how can they be measured in the circulation? No, they can't likely, and we need to have those studies. Multiomics and SNP profile need to be applied on the relevant tissue, normal cancer cells, and to see what's in the circulation. The academias are well suited to explore these avenues, but are nowadays rarely involved in building the registration dossier. Is that an optimal development for the patient and societies? I will pinpoint something which is frequently a problem and missed. Do we have the relevant biobank material which are investigated for correlative therapy effects and patient outcome studies? It's quite well known, in particular for breast cancer, that the primary tumors tend to change during tumor progression. There occur genome phenotypic changes, including that the mutation acquired new mutation. There is ample of literature on that. So it's highly important that you use the correct biopsy material. You should not use primary cancer biopsies if you are studying metastatic disease. Breast cancer guidelines are already informative, but that is not always applied. And we have also found that it's likely so that sequential biopsies together with liquid biopsy may add additional value. It should not unlikely be more ideal to run clinical studies in countries with individual population-based patient registries with complete coverage to capture all relapses, the development of comorbidities, long-term side effects, and cause of death. Unfortunately, the only countries to my knowledge who really have the complete registry for the whole population, these are the Nordic countries, the Netherlands, and UK. Here you see example how the academic world can work with a prospective randomized phase 2 study, which was run a few years back. And here you see the study set up. Here you see how this study was designed, that there were, during the neoadjuvant phase, liquid biopsies were taken, but also repeated biopsy. And we have been using both RNA and DNA-based advanced studies. Immune cells seem to be quite important in how the mechanism of action are run also by endocrine and chemotherapies, question mark to some extent still, but this is an investigational avenue which typically academic groups can deal with, giving highly important information in relation to mechanism of action. Here you see other example. To the left, you have a phase 2 randomized study, which then continued into randomized phase 3 study. And here you see how we have studied the interplay with copy number alteration and immune profiles in early breast cancer. And it has also, we have also studied how, if there are analytical factors involved in genomic analysis working with formalin fixed material. Normally people historically have used frozen material, but apparently formalin fixed tissue can work. Here you see example from a randomized phase 3 study where we're pursuing with the same type of activities with DNA-based sequencing, SNP analysis down to the left corner. And altogether, this highlights that academic environment sometimes potentially can add important value in understanding mechanism of action, difference in side effects, and the heterogeneity is always observed in the clinical studies. And here is just, I would like to express my gratitude to all the patients who have been participating in our different clinical studies, which have been run sometime as academic studies on their own or together with partnership with the pharma industry. And I do think the message is, at least for me, based on my experience in some years, that I think it's a great advantage if the academia can be more thoroughly involved. I've seen the development is going in the other direction, both in Europe and in the United States. And to all of you, thank you for listening to this important meeting, and hope there will be some fruitful discussion at the end, because I think EMA is to really be congratulated for this important workshop. Thank you.
Thank you very much, Jonas. Our last speaker for this session is Professor Christine Desmet, Advancing Cancer Research Through Shared Data and Samples.
Thank you. Thank you very much for the invitation to speak here. It's a great honor. Thanks also, Professor Berg, for sharing your slide in advance. My presentation will actually immediately build further on the previous presentation. So what I'm going to talk about today is actually not only my opinion, but is actually also the opinion of 53 academic clinicians, researchers, and patient advocates we have been working on actually in the past 6 to 9 months. And my partner in crime to coordinate this effort was Professor Hans Wildiers. So we were collaborating with those people principally in the field of breast cancer, as we do breast cancer research. But I think that the principles that we will highlight here really are similar across the field of oncology for other cancer types. And while so far we had a lot of very intensive, very fruitful and constructive discussions here, in a second step, we would really like to open our discussions and the constructive further steps also to pharmaceutical companies. So this is really an open call to please contact me if you want to be involved in the next steps, because we really would like to redefine actually the communication, the balance for biomarker and studies actually in the context of clinical trials, and especially the registration ones. So why is it important? It has already been said several times, but the quality molecular analysis of biospecimens collected from study participants are really essential to identify biomarkers that can tailor treatments to specific subsets of patients who are most likely to benefit. Because we know that it's always only a subset of patients who do have this incremental benefit of a new therapy. And we also know that there is a subset of patients who really have more severe side effects. And it's very important to identify biomarkers for both efficacy and toxicity. And the sharing of data and biospecimens from clinical trials should and really needs to be better in terms of enabling personalized, patient-centric use of cancer therapies and also to accelerate the development of new treatments. And I think that hearing all the presentations before, we already agreed that patients stay in the center of all of us. So it has been already said before, but here maybe documented with a publication. There has been across the year an increasing role of pharma in the coordination of clinical trials in the oncology field. This was a publication from last year in GCO, where they did review over 20,000 trials by clinicaltrial.gov in the US. And then they were looking at the ratio of these clinical trials for industry-sponsored versus federally sponsored trials. And when they looked at the time frame of 2008 until 2012, the ratio was 4.8. And when looking at the next time frame, it was nearly doubling. So obviously, data sharing, when we speak about data, and here not yet by a specimen, this is not something new. And here I'll give some of the things which were obviously already described many years ago. For example, NIH, very early, since 2003, really made data sharing policy mandatory for all new grants and clinical trials which were funded by them. Also, International the Committee on Medical Journal Editors really made it clear that when publishing data from clinical trials, there should be a clear statement on data sharing. And also here, the EMA also mandates the clinical reports with details on methods and results of the trials. However, we see that while in theory this is quite clear, in practice the implementation remains limited, challenging, and sometimes just with pieces of the puzzle. Here, this was a very nice study which was published also very recently in JAMA Oncology, where the authors actually reviewed 91 registration trials in the oncology field, and these were related to 16 different pharma companies. So they could actually they wanted actually to review, to do a kind of meta-analysis to review side effects and outcomes across different ethnic groups and populations. And they were able to get data from 70% of the trials. What they show and they documented is that the sharing of the data could appear and happen through different platforms, with the Vivli platform being used for most of them, the clinical study data request.com being used for another part, the Yale University Open Data Access for some, and then also for others some more proprietary internal platforms which were used. So this looked not so bad, but then actually so the individual participant data could obtain, as I said, for 77% of the trials. And the median time to data provision was 123 days, which is also not so bad. But then actually, when they looked further into what was provided, they saw that there were quite some limitations associated with that, because there were some omission, reduction, or modification of outcome data, or reduction of the assessment variables, of also the non-sharing sometimes of adjustment variables, which precluded to do adjustment analysis, or the lack of study report, or definitions, and so on. So what we did also is that we looked back at the 29 registration trials in the context of breast cancer that led to an approval of the drug in the last 8 years. And what we see here, and I shift a bit to the biospecimen, is that we see that for 28 of the 29 trials, the clinical trial participants were asked to provide tissue and or body fluid biospecimen for biomarker research, and often also at multiple time points. So this really showed and documented that really the patients are increasingly being asked really to provide samples. So but then when we look at the analysis with regard to predictive biomarkers in these registration trials, we we see that actually when the new therapies actually get approval, they are often approved for a broad drug label because of the statistically significant improvements in protocol-defined study endpoints. But as I said at the beginning, the efficacy is often limited to a subset of patients. So when looking back at what has been done actually on these 29 trials, we see that for 29 of these trials, 20 had a published biomarker study, but only mostly one and very, obvious one or a very standard marker. 5 published a predefined biomarker study, but actually no analysis really was conducted that led to a change in indication. So this was from registration trials, and these were also more difficult to access data or samples from, but there are obviously a lot of also successful examples of collaborations, but these are mostly coming from non-registration trials. And these were, for example, in the context of breast cancer, of the assessment of the HER2 status, which is very relevant now for the newer ADCs in some of the trials. Also the association of body mass index, which is also very important. Which is also very important and often not so much studied in the earlier phases, and which is now also studied across other trials. Then also the iSpy2 platform is a very interesting platform, as it is really working with many different companies actually to try to identify those drugs which could be interesting to be taken up in a next phase. And then the Palace trial, which is really also one of the beautiful partnerships between pharma and academia to exploit really biospecimen and data. But so we wanted this to be something, constructive, and this is why we think that, we wanted to come up with 6 points for the way forward. So I think it has been already alluded before, and also by Professor Burke, that there should be, we believe, really like more co-leadership structures in trial design and conduct to make sure that all the needed data is collected. Think, for example, at all the many different trials which did not connect BMI, for example. Also so that prospective biomarker testing beyond the standard markers, because those which are mostly published is according to standard markers, but that these also should be planned. That there should be also biomarker analysis for toxicity planned and prospective validation also in real-world data, and we had already fantastic talks on that before. Also, this has been really, said several times in the previous presentation, the whole of patients should be really more present. When we speak with patient organizations, we realize that, they really very openly consent to donate data samples to support correlative science to develop more tailored treatments for future patients, for the future generation. But they also often assume that their care team, the academic partners, they have been fully involved in the design, that they know exactly what has been going on and what will be done with that, and that it is that it is actually often not the case. So we really think that there should be a greater role for public and patient involvement and engagement here to support effective use of the collected samples and data. And I think that there should be also maybe an explicit mentioning in the informed consent form that clinical and biomarker data should be available for joint biomarker research. Then number 3, also here, the sharing as requirement for regulatory approval. I think that here we see that despite data sharing mechanisms which are in place too often, but I'm not directing to anyone because some are doing really great, but too often pharma typically retain full control of which projects regarding biomarker studies can be approved, which data can be released, which samples can be analyzed, which analyses are done, and also very importantly about the timelines. We also believe that the disclosure of the individual patient data from the registration trials should be encouraged, and also that a plan for data sharing should be present and should be proactively discussed and collaboratively at the time of study design to make sure that everything is very well considered from the beginning, including the platform use, the timelines, and so on. We also think, based on for example, the previous study I showed you, that there is a strong need to standardize the individual patient data packages, really to ensure feasibility also of post-approval meta-analysis, because sometimes these biomarker studies will be underpowered and it will be very critical to merge data from different trials. But this is only possible if the data can be combined also from different studies. Then with regard to the sharing of biospecimens, so far the publications really describing the translational research involving analysis of these patient samples are scarce or often also too much delayed. We have also too many examples, especially in the case of negative trials where actually biospecimens are destroyed or are not available anymore or are not a priority anymore. So I think that the biospecimen custodianship plan should be again developed early with, again, here co-leadership structures. And again, here also there should be a strong need to document and standardize biomarkers assay and interpretation. Then it's not only about clinical data, biospecimen, it's also about the biological data which has been generated. And I think that as soon as it has been generated, it should be shared rapidly, really to avoid duplication and wasting of resource, and also to help in the design of new clinical trials. It could be shared via recognized platforms, but here again also standardization and documentation is needed. So these were some of the points which we obviously describe in many more details in our publication, but we think that this can be really beneficial for all stakeholders, because we believe that having a better transparency and a better use of these samples can really boost patient engagement, can also build further public trust to enhance credibility. We believe that it can drive joint innovation. We also believe that it improves really scientific credibility on all sides, that it can also influence policy, that it can also optimize the use of the different drugs to really focus on the subpopulations who will really have the meaningful benefit, that it can advance science, and that it can also appeal to other investors. And so again, I want to thank all the different collaborators, and some are in the room here. And I thank you for your attention. And as I said before, in the next phase, we are very willing to work with some of you, pharma collaborators representatives. So please reach out if you're interested. Thank you.
Thank you very much, Christine and both speakers for very clear presentations. Clearly lots of interest also from patient organization on this sharing of data and tissue. Lots of opportunities, and also, as we've heard from Professor Berg, capacity from academic researchers to develop activities in this area. So something to come back to. We're now moving to the last part of this workshop, which is our panel discussion. I'd like you, we have highlighted some of you to be sort of speakers in the panel, so I'll call upon you in a certain order to say something, but eventually I want you to come back to, are there concrete initiatives that we can bring forward as industry and the Cancer Medicines Forum and EMA together? Is there a willingness to do so? And how EMA can support this? But before that, I'd like to hear from our panelists, and I'm going to slightly reverse the order because I heard Pierre needs to leave. Pierre Demolis, he's our Oncology Working Party chair. Pierre, you've heard a lot. What are your thoughts about these challenges and opportunities?
Thanks, Francesco. You know me, you know my limits. So I will go for very simple-minded comments. If you want to optimize a treatment, optimization means that the treatment is on the market. So the first condition is a marketing authorization. If you want a marketing authorization, you must have established that the benefit risk is positive. We are in the domain of cancer. our culture in cancer comes from the time where we had toxic chemotherapies. So everything was on the efficacy and the safety, who cares? So we want to be efficient, whichever the price we have to pay on safety. We are far from this system. We now have modern treatments that are closer to what we can observe in other therapeutic areas, but still we have that culture that if we want a marketing authorization, we must be sure that we demonstrate some efficacy. And if the safety is not optimal optimization, you follow me, if the efficacy is not optimal, however, we can get a marketing authorization. And then the whole question is, once we have the marketing authorization, are we sure that we treat the right patients at the right dose with the right combinations for the right time? And some of these questions can be left unanswered at the time of the marketing authorization. I will take an example. Nafsika and Denis spoke of elderly patients, because optimization is not only optimization of the dose, it could be optimization of the population. If you look at the trials that allow registration of products for multiple myeloma, you suddenly observe a striking difference. The population is much younger than the population that will be treated in reality. This is not normal. Why that? If I want to develop a drug in multiple myeloma, I want to be sure that I get a marketing authorization. The efficacy must be roughly the same, whichever the age. It's not the problem. The safety is the problem. So if I do not pay much attention to the age of the patients, and the enrollment results in much younger patients, I have the best conditions to observe efficacy and the best conditions not to get embarrassing safety problems. If my drug is very, very efficient, it will be used in the elderly, however, even if we do not have information. And if the benefit risk is borderline, I will be very happy that I have not included elderly patients, because with the safety problems, they would have transformed the benefit-risk balance into something which is highly disputable. What I'm saying is that there is no interest in developing the drug the best way, and this is not accusing anyone. This is how the world works. And if we want people to develop drugs the right way, with the help of academics, of patients, of anyone, and of the agencies, there must be incentives. So what is the solution? We know that we are in a closed system where the money is not something which is infinite. We have to pay for the incentives. So we are not here at the EMA in the driving seat for organizing a global reflection on how much we can pay for innovative drugs, how much we can pay for the incentives to develop the drug properly, because I believe that incentives are probably the only solution. And then, with the money, we will find a solution to organize transversal plans to get better information on elderly patients, on the right dose, on the right combination, on the right timing. So my conclusion would be that we cannot decide anything that will improve the system here between us. We can only speak to higher people. It could be at the level of the Commission, it could be at the level of the national authorities. I believe much more in the European system, to have a global reflection. And what is obstacle to that global reflection? It is the fact that the system is completely puzzled. We have the payers, and consider that the payers are different in every country. We have people justifying how we spend money. This is the responsibility of each government. We have the European Commission and the European power edicting rules, but those rules are not necessarily observed everywhere, because we have the possibility for national adaptations. We have the hospitals, each one with his system. We have the reimbursement systems that are completely different. We have all the decisions taken at different levels, not speaking to each other, to organize something which is globally pushing toward the proper development of drugs. So it could be seen as pessimistic as a conclusion. I think that if we want a solution, the solution must come from upper.
Pierre, thanks. This is very insightful, and I don't think it's pessimistic at all, and it does not negate the fact that to draw attention on these problems, we can have a role, but we cannot we're not empowered to enact solutions that involve financing of trials and things like that. All right, thanks a lot, Pierre. Colleagues in the panel, I'm going to go through the list and perhaps start by the people who have not given a presentation yet. So, Francis, forgive me. I'm going to ask Pavan Vajja first for a comment.
Yep. Thank you. I think this is a really nice forum. I think I'm a clinical pharmacologist by training, so I come from what Chunzi Lee and others have presented, so that's my area of expertise. But what was really interesting is dose optimization means something different for everyone. I think that's what I'm seeing here. Like I think, Sahar, your presentation where you are thinking about having drugs in the neoadjuvant versus adjuvant setting, that's dose optimization for you. So, but in a traditional dose optimization sense from a drug development perspective, I think with the Project Optimus from FDA and initiatives, and this is, I guess, will be an initiative in that direction, I think we are doing reasonably well for dose optimization. I think if I think about the last few products which I worked on from AstraZeneca, we are running randomized trials in order to optimize the dose. So I think a move in that direction has already started. I think the opportunity from what I'm hearing from all of us is, can we leverage the data which is there in the real world evidence? Can we leverage some of the innovative techniques like the biosamples as well as some of the in vitro humanized models? Can we use the data from the first indication dose optimization to the next indications rather than subjecting patients to clinical trials for at lower doses in different indications? So I think that is one of the things which I think we as a community can have as an objective. Can we do dose optimization for the first indication? Then can we extrapolate that data using all the modeling approaches, data that is being generated in real world evidence to support the dose optimization for upcoming trials or upcoming indications? There is a role of dose optimization post-registration, I think, specifically in some subpopulations. Like I think we discussed geriatric patient population or things like that. I think the pediatric dose optimization is much more well established, but I think that is geriatric patient population is something which can be explored. So that's where I would stop. I think there are opportunities there.
Thanks, and I appreciate that there are different priorities also in terms of what research questions to tackle and with what evidence. And I think that's something we could definitely discuss because I sense we're not on the same page sometimes there. I'm going to jump to Professor Dirk Arnold. Professor Arnold, any immediate comments?
No, thank you. And I do have the pleasure to speak here on behalf of for ESMO. And I do have to, let's say, congratulate you for this excellent forum with the many aspects we have been discussing and we have been learning. ESMO is a society which is committed clearly to distribute the best available drugs or treatments to patients. And we also do feel responsibility for healthcare environments, we'll say, global oncology situation where it has to do with the access to drugs. And when we come to the interface of clinical trials and the collaborative mode with the industry as another stakeholder, I would just like to add an aspect which has not yet been mentioned this afternoon, and maybe we've spent some time in this forum now also. This is also those optimization has been a topic, but in general, de-escalation and treatment shortening, treatment optimization by limiting treatment intensity, etc., is a way for, is done in many academic societies, in many academic environments. And as we as ESMO have to handle the value of trials, and as we do, as you do know from with instruments like the magnitude of clinical benefit scale, etc., where we put the treating physicians and researchers' perspective to the value, we would like to also to, let's say, enlighten that also this kind of trials could or hopefully will prompt into collaborative efforts that not every label, which is, let's say, the label by the moment of registration, will be the best label for the best patient or environmental interest. So therefore, I would love to add this also to this panel discussion, because I think also for this type of trials, we are really seeking for a good collaborative effort.
Thank you. Very, very clear. Colleagues from the panel, I'm just going to continue on the list, but please feel free to react. So jumping back to industry, Liz Poole. Don't know if Liz is...
Hello.
Yes.
Yep, I am here. Thank you. This was a really, really wonderful set of presentations, and I took some notes during the presentations and had a few areas that I wanted to touch on. First, I should say I represent Jazz Pharmaceuticals, and I lead our real-world evidence team as long as many of our other post-marketing activities. So I wanted to touch on a couple of things. In Nafsika's presentation, which was lovely, she talked about more not always being more or better. And I think that's a really interesting topic to think about. What needs to be in clinical trials and what can be saved for the post-marketing phase. So, particularly with that dose optimization hat on, or that framework, how do we think about what could be done in the post-marketing setting and what should be done in the post-marketing setting once the drugs are available to a wider range of patients? So I think that's a topic worth spending some more time on. I also wanted to talk about real-world evidence having come up in multiple places here, and thinking about the acceptability of RWE to regulators and how regulators might help industry push to get real data on real patients, including how the drug is actually being used in the real world for these patients that haven't been studied in the clinical trials, which has come up a number of times over the course of these presentations. And I think Pavan already mentioned this in his comments, but I think we have a lot of really good examples from pediatric cancers where everyone agrees we need to get this right, and we have a good system in place for dose optimization. And what can we learn from those? In particular, I think there's a long and healthy sense of collaboration between industry and academic groups for pediatric cancers, where the benefits and the incentives to everyone are clear, to really make sure that we're getting dose optimized, both for efficacy and safety. But also there are a lot of good incentives to industry to work with these groups, particularly in pediatric cancers, to think about how do we do the best we can for patients, and how do we make sure that we can do things like de-escalation and fewer treatments. So I think there's a lot of really good topics that came up here today that we could dig into more, and it was just really enjoyable to hear everyone's take. So thank you very much for including me and for allowing me to reflect on a few of those pieces.
Thank you, Elizabeth. And continuing in the panel, Marike Schonen.
Thank you, Francesco, and thank you very much for the invitation to serve on the panel. I'm here to represent UCOPE. I'm an epidemiologist at Amgen. So building on the comments made by others, there's one thing I wanted to pick up on. We've heard a lot about clinical trials today, and of course my passion lies with real-world data. And one of the things that occurred to me is that there seem to be a lot of areas where real-world data can also serve across the development life cycle, not just to complement once a cancer medicine is on the market, but are there areas where real-world evidence can be used to optimize the knowledge around the disease, where very early very early on patient populations can be described in further detail, such that we understand what is their profile, what are the adverse events that we can expect in this patient population. So on that note, just some thoughts that I wanted to add and complement the other speakers. But I feel that there is huge opportunity for in the real-world evidence space to have close collaborations with academia, also with the regulator and industry, to really think about where can we help medicines development. That is particularly pertinent probably in the novel biomarker space, where no one really knows. So can we all come together and collaborate to help better understand that situation? And I think the key there really is the trust and the transparency. And certainly, in the real-world evidence space, where data is already existing, and oftentimes, academic groups, maybe hospitals, have their own, perhaps smaller scale registries where there's very deep knowledge of the disease and the patient population, but then there's equally deep knowledge and understanding of perhaps the technical aspects of using the relevant methodology to help analyze the data that lies within industry. And certainly, personally, I've worked on a number of examples where that close collaboration was extremely positive, productive, and ultimately really led to being able to make the right decisions for the patients. So that's one thing to be mindful of, and really also picking up on the point, Francesco, you made much earlier on in the day about being clear what questions to answer when in the development plan, and I really feel that real data has definitely got a place there. And then having said that, one thing to really bear in mind, which is a much more broader position, outside of even the optimization of cancer medicines, but is valid for anything to do with real-world data. We have to be very careful that we use fit-for-purpose data when we try to answer questions, that we are confident that the quality of the data is appropriate. And then, I would also say, in the real data space, of course, the European health data space is a huge opportunity where we can also think about opportunities for data sharing, and very keen on seeing how that evolves. Maybe one final comment then around sort of the methodology piece that I alluded to a little bit earlier. We are extremely thankful for the various guidance documents that have been developed recently and are some still out for public consultation. External comparator, patient experience data, all these things are really going to help us collectively to ensure that we, collect the relevant data to help inform cancer medicines development. So I can keep talking, but I'll leave it at that. Thank you.
Thank you. Tarek, you've been involved in a number of real-world data initiatives. Is that the solution to at least part of our treatment optimization conundrums?
To answer that, I think it maybe is the solution to identify where we need, particularly to do studies. I'm not sure that we can really understand treatment optimization fully from real-world data. There's so much confounding in these things, but it definitely will tell us how it's used in the real-world setting. So I speak for hematology today, and I haven't prepared a long speech, I would say, but one thing I want to say is that the only reason we're discussing this is because it's been a huge success, cancer treatment over many years, speaking for hematology at least. Like, we have so many diseases now that are chronic diseases where we occasionally believe that the residual life expectancy becomes very close to normal. And that, of course, raises a lot of other topics, like how long should we treat them, how heavily should they be treated, how should we weigh side effects against efficacy and all these things. And this is really where we are challenged today. And what we kind of can get a bit concerned of as clinicians in hematology, I would say, is these kinds of labels that says treatment until unacceptable toxicity, for example, because we know in reality that many patients do have some side effects, and many patients are in situations where they are in remission for long time, maybe a couple of years, and it wouldn't take much for me to be convinced that if there are slight impact on quality of life or side effects, such patients may be better off having a treatment holiday. So this is really some research that is needed, not only the dose, but also to support what was said from ESMO, to look at the duration of treatment. And especially also how we get these trials that academia is probably going to be responsible for doing into a quality that could lead to a change in label, for example, so that we get it all right. Yeah. I think the big hurdle, of course, is how do we collaborate in these things, academia and pharma, and how do we fund these things? Because there are so many different interests, and I'm not going to say anything that can solve this today. But I think this is going to be a major thing in moving forward. Like how do we align all the different interests in something that is a shared goal? Beyond that, we all want the best for the patients, but there are also different interests that are kind of diverging a bit occasionally. Yeah.
Thank you. And I've seen Professor Gisselbrecht has raised his hand. I'm just going to check, Professor Gisselbrecht, just 1 second to finish. Francis, rapidly, and Denis, a few words before we open also to Professor Gisselbrecht. Francis.
Thank you very much. So I will keep it very short because I've already had the opportunity to speak, of course, but I would like to say that it's very useful to see all these different perspectives and to be able to also understand from each side, I mean, where really the priorities lie. And so yeah, of course, I'm also more on the real evidence side of things. So I think it's very important to keep this dialogue going. And so yeah, to make sure that we have the right incentives for all the post-marketing research and keep the dialogue going. Thank you.
Thank you. And Denis Lacombe.
Thank you. I think for me it's a very important day because actually our first publication about rethinking the models of development was in 2013. And I guess we were the only one to speak about treatment optimization at this time, and it took about 5 years until we convinced the European Parliament to work with us, and we published a manifesto in 2018. And then that helped us to go a bit further, and this is in 22 indeed that we started the Cancer Medicine Forum. So it's been a 12-year trajectory today. So I think it's a big achievement. We are not there. Because the ambition is high. We want certainly to deliver a robust prospective data set on which we can comfortably say to patients how we are going to treat them. Indeed, it's not only about those. The trial I showed is a schedule. Question, and as Pierre said, we have new challenges, so we really have to re-engineer and potentially to see what is the role of the different stakeholders. 20 years ago, 10 years ago, academia was not what it is today. I think there are important societal questions that the independent sector can be mandated to take. There will be some that we can do in partnership with industry, some that we will have to do alone. I think what is important is that we maintain constantly learning healthcare system. There are clinical questions in care that we need to take and to answer, and we have to make sure that we all push in the direction that research also should be conducted into care. Otherwise, I believe that the programs that we have are at risk to be insufficient to make inroads for, I would say, progressing on cancer outcome. I think there are challenges also because that's what Pierre said, where to take the decision, and EMA has been instrumental. We are certainly facing the challenge of what is a supranational competence and what is a national competence. So I think we still need to bring the healthcare system with us, and potentially this is the next stage we have to take. Thank you.
Thank you, Denis. Professor Gisselbrecht, and then Philip Josephson.
Yes, thank you very much. I think Denis explained the situation. In fact, at the present time, when you are a clinician treating patients, you are one set of package with biological marker, clinical data, and so on and so forth. So that's fine. Nevertheless, it's not so easy to put all the clinicians together or all the study together. And that's one of the main criticisms that we have on the way we are all working at the present time. Not for a personal reason, but because we are changing the technology. If you look at the hematology, how the different tests are different from 1 year to 10 years later. And we have really to make pressure, or the authority has to make pressure to allow us to have a connection with the data and to be sure that the ethical committee, for example, in one country will say, okay, you cannot do that and so on, or you have to do again all the procedure. So I think that's my main concern. We made progress in the past, but when I see all the new entity, new marker, and new treatment, I think it's something to do at the present time.
Thank you. Philip Josephson, can you introduce yourself?
Yeah, thanks. I'm Philip Josephson. I'm a regulator from Sweden working within the EMA system. I'd like to pick up a little bit on what you said, Tarek, about de-escalation or de-intensification studies and how to make sure they are fit to inform the label, because this is something we have examples where the community has decided on less intensive therapies, but the data have not been considered to be sufficient to inform labeling changes, rightly or wrongly. But a lot of this conversation has to do with there will be some therapies where there's going to be an interest after having established efficacy and a positive benefit risk. There's going to be an interest in whether the same treatment goals can be achieved with simply a less drug and better tolerability. So here you have essentially a scientific equivalence question. You're wanting to show that you can have this less intense therapy while still having something that is effectively understood as similarly good. And I'm not sure we have very good regulatory guidance, and I'm not sure we even have a very, I think maybe there should be more thinking about, in such context, how do you establish that equivalence? Because when we've had experiences of discussing such study designs with sponsors, it very easily becomes that the study size and duration becomes prohibitive. So these kind of discussions are both sort of what is the acceptable uncertainty about this equivalence, and also what is the role of earlier endpoint, for instance, objective responses or complete responses or PFS or whatever. What are the endpoints that are required to be similar in order to sort of infer that the entire outcome, including, for instance, overall survival, would be similar? I think that perhaps carrying on that conversation between stakeholders would be something that could come out of this meeting.
Colleagues, I see a lot of hands online. Please, let's keep our comments short so that everybody can have a say. Karine Cohen, please could you introduce yourself and tell us your comment? Karine Cohen? If not...
Sorry, thank you, sorry. So I'm a patient, also a professional, but I speak here only as a patient. I'd like to say that I was absolutely distressed to see that in the area of early breast cancer, the same criteria that I was used to, over drugs are not applied. Like, for example, in 2020, possibly because of a COVID crisis, that's what I would like to believe, there's been a big discrepancy in the assessment of adjuvant abemaciclib between EMA and FDA. They didn't align on the number of grade 5 events. That's something around 20 by the FDA, and they didn't disclose their regulatory file, but one can read in the label 0.8% in a population that is mostly cured, right? And the EMA, they said 1 or 2 possibly fatal adverse events attributed to the drug. And this impairs the benefit risk, of course. Furthermore, since most of these are cardiovascular events that continue to accrue over time, and even more so in the real life population where people are not as young and as fit as in the trials. And there's no follow-up on that either, in a real-world analysis, neither in Monarch E trial, where amazingly in 2024, there's been a publication by Götz that shows that the less drug you take, the better outcomes you have. And this is with acquired resistance, another issue with this drug. And I'm asking to get these curves updated because now they have 7 years post of follow-up, and don't get that curves, because of course it doesn't work. Plus there's a big controversy where people are attacking them as well as Novartis, of course, as they did the trial in countries that had no access to post-treatment standard, post-progression standard drugs in case of metastatic disease, and this impairs their overall survival. And normally it's a job of authorities to get this data out because you don't release a drug without follow-up on so massive grade 5 events and so important questions that's been raised then in the HTE, in country level at the HTE. They ask what do we give as post-progression treatments. And so I see this dysfunctionings here. I understand it's a lot political, but I like everybody to hear that. And I hope it's going to get fixed sooner rather than later because there are thousands of people take these drugs. Okay?
Thank you very much for your comment. Monica Rakovita.
Hello, everyone. Thank you very much for having me. I'm an access and policy manager with Myeloma Patients Europe, an umbrella organization that represents patients with a rare blood cancer, multiple myeloma. I've been through some very interesting presentations and very, very interesting issues in this workshop. But I must say, it was a bit striking for me to notice that among all the stakeholders mentioned as possible collaborators and key stakeholders in a healthcare ecosystem, patients to be left out. And that is we are in an ongoing and an open consultation by email on a reflection paper on patient experience data, where it said that one of the key factors of a successful treatment is including patient expectations, preferences, and values. And in a wider context of regulatory agencies all over the world are increasingly involving patient representatives, sometimes at the level of decision making. And of course, I'm mentioning UHTA and then various HTA agencies throughout Europe. So I wanted to remind everyone that patients are not just as passive receivers of treatment. They can be very valuable collaborators, active participants, up to the point of co-designing clinical trials. And that it's not making such trials more relevant, effective, and patient-friendly. But that can help address practical issues from the very beginning, therefore saving time and valuable resources. That would lead to an improvement in recruitment and retention, which are still very big issues affecting all clinical trials. Would lead also to a user-centered trial design and materials such as consent forms. So overall, please do keep in mind patient representatives, patient organizations, patient groups, patient experts, because a lot of the patients are not just able to speak about their personal experience with the disease. A lot of them have become, over the years, quite experts in their particular disease or their particular area, medical area I mentioned. And some of them have extensive experience being involved in regulatory frameworks. So please do keep us in mind. Thank you very much.
Thank you, Monica. Very important comments. Professor Arnold, and then Tarek.
Yeah, and just reframing what has been commented also by my colleague from EHA, Professor Galali, and also thank you for the voice from EMA for this. I personally do think that exactly what has been, let's say, coming up, that is kind of compromise of the accepted level of non-uncertainty, which we have to accept and balance versus the gain, let's say, for toxicity aspects from the patient side as well as from the health economical aspects, is clearly the thing which affects many clinical trial designs or drives many clinical trial designs from academic trials, and which is asked by health societies as well as from patients. So therefore, I think this kind of collaborative spirit is to be, let's say, developed, and also methodologically what we are really assessing, what we are really accepting as potential non-inferiority level is really, which is on the topic. Of course, we can always make the answer simple and can say, well, if non-inferiority trials which are designed are negative, then it's not acceptable. And we all know that this is not the right answer to this, because we will never end up in this world of non-inferiority trials, which will prove every point of de-escalation. So in essence, I would love to let's say, keep on this track and develop with all of the stakeholders here, maybe in a future forum on this methodology.
Thank you, Professor Arnold. Tarek, last word from the panel, and then we're going to wrap up.
So just a short comment to another type of treatment that should be discussed in this setting is the sequencing of treatment. So we have quite a lot of therapies that are tested as triplets or maybe even more or less. And they always give a nice PFS benefit, but often no OS benefit. And really what would be clinically useful to know here is if the sequence, like giving the drugs in a sequence, is as good as giving everything up front. And I also see here there's, of course, a conflict between interests, but this is really something we need to have much more knowledge about.
Thank you, Tarek. Thank you, colleagues. This has been an excellent discussion. I came into this meeting asking myself, can we continue this conversation? And I think the answer is overwhelmingly yes. There is a lot of interest, a lot of proposal for topics that we could develop together. I think we've all realized, and Monica reminded us, that this is not something that any stakeholder on their own can bring forward. It's something we need to work on all together, and the Cancer Medicines Forum could be a good, let's say, start for this type of multi-stakeholder discussions. Whether we will achieve anything very concrete or not, that we cannot promise, but at least we can promise that we will focus people's attention on what we think are shared priorities or the priorities of the different stakeholders. Denis, since you didn't have to leave, I'm going to leave you the final word here. You have created the idea for the Cancer Medicine Forum, so I think you should be punished here with the last word, and then we close the meeting.
No, thank you, Francesco. You are putting the bar very high. No, I really, again, think that we have to rethink our models. I like the idea here about especially doing research on research methodology, because indeed we have to see how we are going to do these trials. I think it's very important that we see a will in changing things, and again, indeed, the starting point and the end point is the patient. And again, I would just want to re-emphasize our learning curve on the de-escalate prostate cancer trial, where it was really a nice experience to design a non-inferiority clinical trial with all the limitations it has and all the challenges it has, but with the patients about how you design that, what is the acceptability, knowing that it's not perfect. But we are trying to correct something that is important, because today it's a daily question, and doctors don't know what to do. So basically, it's basically a matter of discussion on day-to-day, patient per patient, and we cannot tolerate this uncertainty.
Thank you, Denis. So, colleagues, before closing, what I want to do is that I'm going to work with Fabio Borges from ERTC to try to summarize the discussion and work with all the speakers here to try to come up with a few points that we could work on further, and then we can discuss whether you guys are and we are committed to have perhaps some groups to discuss topics further and advance like this. Okay, so if that's okay for everyone, then I only have to thank all the speakers, all the panelists, all participants, colleagues who connected. Thank you all very much, and have a great evening.